we can thought on transplant immunology in two different fields, depending on the type of graft that is involved. So in the case of vascularized solid organs, the more general simplistic view is just to think that we are introducing with the graft some antigen presenting cells and also endothelium that will be in contact with the immune system of the recipient, and all the alloreactive reactions will take place with these two main type of populations in the front line. On the opposite, when a bone marrow transplant is performed, there is a conditioning of the patient, and we are really doing a transplant, introducing a new whole immune system in the recipient. And it had traditionally has been evolved as two different fields and managed with different teams and follow-up programs. And what I will try to introduce or try to discuss in this talk is that there are different situations in between depending on the type of graft, and there are different pathways that should be considered. From also in this very simplistic approach, we can characterize the rejection process in different classifications. So for vascularized solid organs, normally we think on hyperacute, acute, or chronic rejection, especially related to the dynamics, and then we will see that there are different pathways, immune pathways that can be involved in each of them. And in bone marrow transplant, the most important immunologic reaction is the graft versus host disease. That is a kind of immune attack of the immune cells that are elicited with the bone marrow transplant against the host cells. One of the very important initial concepts that we need to have here is that alloreactive response is really a conventional immune response that has some specific rules, but we always need a presentation event, activation of lymphocytes, amplification, effector differentiation. So normally this has the kinetics of an immune response in which we need some days from the initial allorecognition events to the full activation of the effector mechanisms. And this kinetic is can also be seen in the clinic, depending on, for sure, on the different type of situation that we will then discuss a bit more into deep, but also on the immunosuppressive regime that is used. It's important to be considered which are the different allorecognition pathways that can activate the immune response. So there are different pathways. The direct allorecognition pathway has been very recognized in the very initial characterization of solid organ transplantation as the primary mechanism of cellular rejection, in which a donor antigen presenting cell with, in the context of the donor HLA system, activates recipient T cells by a direct allorecognition pathway in which there is a peptide that is presented, but also there is a huge cross-reactivity or huge alloreactivity in the recipient compartment, because this MHC has not been used for timing education of the recipient T cells, and this is a pathway that contributes to acute rejection and activation of different T cell population that may be CD4 or CD8 T cells, depending on the type of MHC that is recognized. But there is also another possibility that is the indirect pathway, in which normally structural cells on the solid organ that are from donor origin that persist long time after transplantation. And even if there is a replacement of the donor antigen presenting cells by recipient antigen presenting cells in the either in the graft or in the rest of the organism, there is a persistence of allopeptides or alloproteins, especially the MHC of the donor, that can be introduced to different antigen-presenting pathways on recipient cells and just presenting donor-derived MHC peptides that activate recipient T cells. And this is something that may contribute to rejection in different moments at the initial times, but also in the long term. And it's one of the drivers of the chronic rejections. And this may happen, this may happen mainly by presentation of the extrinsic pathway in the class 2 context, but in some cases also cross-presentation can also introduce other type of reaction, but it's mainly a CD4-driven response, and it may sustain the appearance of donor-specific antibodies in the long term. And there is another pathway, is the semi-direct pathway that has been recognized just from now, almost around 20 years before, in which there is the possibility of the donor antigen presenting, the recipient antigen presenting cells that incorporate just macrovesicles containing the full MHC of the donor, and you incorporate this in the membrane, and let me see. And then they can interact with the recipient lymphocytes and perform an activation, but just cross-dressing with the donor MHC, the recipient antigen-presenting cells, and this can also trigger allogenic reaction in different kinetics at the very beginning, but this is also a pathway that continues several years after the graft is in place. So these three pathways have been recognized as different drivers of allogenic reactions at with different intensity at different times after transplantation. And one of the important things or other important concepts is that the frequency of the alloreactive species is much higher than the physiological frequency of lymphocyte activation at a foreign antigen, especially when the full MHC molecule is involved. So the direct and the semi-direct allorecognition pathways, they have a much higher frequency of activation of lymphocytes, and that's why the allogenic response is very intense, and we need to have immunosuppressive regimes after solid organ transplantation to prevent this. But also the indirect pathway that has a much lower response is a pathway that prolongs or lasts several years, and it's always a threat. So that's why normally immunosuppressive regime after solid organ transplantation is not suspended. And in the recent years, there has been a fourth pathway that has been recognized. This is important in different type of transplantation, that is the inverted direct pathway. How this works? This is in the case in which the solid organs introduce also donor CD4 cells, even in the case that organized lymphoid structures are part of the graft. One possibility is that there is a direct interaction between recipient B cells and donor CD4 cells, and this interaction activates the generation of donor-specific antibodies by this interaction of the allogenic interaction of the full MHC in the just mimicking the direct pathway, but in the opposite way. So just they are the donor CD4 cells that are the ones that are activated by this allogenic reaction and cooperate with the recipient B cells to generate a donor-specific antibody response. This has been recently shown in a very elegant paper in which they show that even using recipient in experiments using recipient animals that have no CD4 cell component, but using a graft that introduce with a CD4 normal phenotype, this CD4 that come into the graft that can originate the donor-specific antibodies. And if you consider what's going on in the clinics, these are data from different type of grafts in the clinic. So normally, grafts that are very poor in CD4 cells, such as kidney transplantation, the kinetic of regeneration of DSA is much lower. But in the case of other grafts, such as lung or even worse, intestine, in which there are a lot of organized lymphoid structures that are transplanted, there is a kinetic of rise of donor-specific antibodies that is much faster. So this is another pathway that should be controlled to avoid the appearance of donor-specific antibodies. So all these 4 different pathways should or can be operating at the same time after solid organ transplantation, and they are the ones that have to be controlled by immunosuppressive regime. But there is also another very important thing is that alloreactive response can also be primary or secondary, such as different normal immune responses. So there are 2 different, very, very different situations in which there is naive recipient in which there are no pre-existing alloreactivity, so the kinetics and the intensity of the response would be such as a primary response. But in the case that patients are sensitized, even with the presence of antibodies or reactive cells previous to transplant, this is a very harder scenario. So the determination of pre-existing alloreactivity is critical. These are the things that are exploring the characterization of pre-transplant in the pre-transplant period, and normally the cross-match analysis is performed to detect pre-existing antibodies and take measures such as doing some desensitization or even avoiding or the use of specific organ in the case that there is a high alloreactivity. And well, we will discuss much more about this type of procedures in the course. But the important thing is that, or the important concept is that to retain is that we can have this primary or secondary alloreactive response. And so, which is the origin of this alloreactivity? So normally there are different explanation or situations that can trigger alloreactive. So the use of transfusions, even in the case of using just erythrocytes, there are some minor antigens that can trigger some alloreactivity. In the case of platelet transfusions, there is a HLA sensitization. Previous pregnancies, in the case of adult patients, that can trigger also specific HLA recognition. And there is also a possibility of having some molecular mimicry between the specific HLA alleles that can also trigger a reactive response by this cross-reactivity. And for sure that the biggest challenge is the previous organ transplantation. So in the case of patients that receive in different moments a second or even a third transplant, this is the the survival of the graft in this situation. So normally there is much higher acute rejection and also higher chronic rejection in the case of pre-existing alloreactive response. So this is something that should be considered, and also is something that is surveyed before doing the transplantation to condition the patients. So another very important thing is that in every solid organ transplantation, the procedure involves the organ procurement, but also the preservation, that there are different ways of preserving organs, and now there are many alternatives going on into even reaching the clinics on dynamic preservation. But in any case, the implantation involves the reestablishing the circulation, but there is always ischemia reperfusion injury. So in the setting of a transplantation, we already face not only a reactive response, but also inflammatory response at the moment of the transplantation because of the ischemia reperfusion injury. And this is something that normally a stronger immunosuppression is used at the moment of transplantation to just mitigate this environment. And so after having this general concept set, another thing that I want to introduce is the two ways alloreactive transplantation pathway. So normally we can also have the situations in which the graft has some immune cells, and normally there is a two ways circulation of graft immune cells that go into the recipient, and also recipient immune cells, either antigen presenting cells or lymphocytes that cross circulate in between the graft and the recipient. And this may depend for sure on the type of graft that is used. But in both directions, alloreactive response may take place and expand either donor-derived lymphocytes or recipient-derived lymphocytes, and this may trigger rejection in the case of expansion of recipient lymphocytes, and also graft-versus-host disease in the case of transplant or graft activation of graft lymphocytes. So these two ways alloreactive response is going on in different situations, and what I will show today is some examples on just visceral organ transplantation, mainly intestine and multivisceral, in which this situation can have, or they can take place both at the same time. So in a very simplistic also way of thinking, we have this fight against donor and recipient lymphocytes. And if there is just an over response or an overexpansion of recipient lymphocytes, we will have rejection reactions. And in the case of intestinal transplantation, this is one of the most frequent complications, being very frequent in the clinical setting. But on the other way around, if there is an expansion of donor lymphocytes activated and that can overpass a certain threshold of clinical significance, we can have also, in this case, graft-versus-host disease. And so some in solid organ transplantation, either the isolated intestine and the multivisceral transplantation may have graft-versus-host disease as a complication, and this is very important or has a big impact in the clinic, because it's very hard to treat, and we need to learn more on how to deal with this situation. But what is important is that this two-way LR activity is present even in the case, even in without clinical symptoms. So this is a very interesting paper from the group of Megan Sykes and Shannon Few in New York. So they just using HLA mismatch and the combination of the antibodies, they can detect donor-derived myeloid cells, and they can show that at the different compartments, there is a multilineage chimerism in the case of intestinal transplant patients, even without clinical complications such as rejection of graft versus host disease. So, mm, in the case of either lymphoid myeloid cells, we can have this. And what is interesting is that they can show that several months after transplantation, there is the donor-derived lymphocyte tolerate the receptor. So in some way, alloreactive response can be controlled in this scenario. So this is very interesting to try to understand also how we can deal with tolerance and how we can use this for the well-being of the patients. Also, they show that these different lineages are originated by hematopoietic precursors that are present either in the intestine or in the liver of the graft that they use, and that these precursors are functional equivalent to the bone marrow derived, like they can originate the different lineages. So with the different evidence gathered in this paper and also in other publications along the last 5 years, we can have these models of repopulation and two-way activity complete picture in this type of when the grafts have enough presence of precursors, and even in these cases they show that donor derived precursor can populate the bone marrow and originate a stable chimeric state, and this without any type of clinical complication. So there are several questions around how this situation can be used and can be promoted to just release immunosuppression and establish a kind of tolerance state in the patients. And I will show some results in the clinics that how we in the clinic in our experimental setting on how we can deal with that. But so far what we have just go along is that alloreactive response depends on the frequency of the allorecognition events, but this follows the dynamics of a conventional immune response. And this two-way alloreactive paradigm predicts multiple interactions. This depends on the type of graft involved. So this is a bit more complicated than the initial picture that we discussed, in which bone marrow transplant and solid organ are two different situations. We have a kind of continuum depending on the presence of precursors, hematopoietic precursors in the grafts that are transplanted. So liver, intestine, multivisceral, and even in lung, this two-way alloreactive reactivity may lead to more complicated scenarios than the previously considered. So we have set up some animal models to study this paradigm. And in the case of multivisceral transplantation, we are very interested in understanding the problematic of graft versus host disease. And so we established a microsurgical model in rats, mimicking the multivisual transplantation that is done in the clinics. So the graft consists of the full GI tract from the stomach to the colon, and this modified multivisual normally includes also the spleen. And this is something that there is a big controversy in which what is better if we can just keep the spleen in the graft, because this is from the anatomic point of view and the surgical point of view is much simpler, or to just separate the spleen of the graft, which is more risky and more complicated on the technical point of view. So normally our model is an heterotopic, so we use vascular anastomosis, um, the the small mesenteric artery to the aorta, and also the porto cava anastomosis. And in our case, we wanted to contribute to this surgical controversy, trying to show in which situation graft-versus-host disease may take place, depending on the anatomy of the graft and the anatomy of the recipient. So many surgical teams, they use a recipient splenectomy just to generate more space for the graft, but this may also imbalance this donor and graft lymphocyte load. So in our settings, we perform two different type of groups. So isogenic and allogenic multivisceral transplantation. And we compare also groups in which the recipient has been splenectomized, to understand if this may affect the frequency of graft-versus-host disease appearance, that this is something that has been very discussed in the clinics. So we could observe in our model that in the 4 to 6 days after transplantation, there is a rash, and this mimics the graft-versus-host disease, and we also identify this donor-derived cells here, and could confirm that this is a typical graft-versus-host disease, which has not been shown before in solid organ transplantation experimental models. And we also, comparing both groups, we observe graft-versus-host disease in all the animals that a recipient has been splenectomized, whereas in the other groups, we have a mixed reaction. And we also could track the donor and recipient lymphocytes in these animals. And what we observe is that in the case that there is an splenectomy in the recipient, we have a much higher chimerism, and this fits with this paradigm of enhanced predomination of activation of donor-derived lymphocytes. And in the case of no splenectomy, we observe that at later time points, disappearance of the donor-derived cells and also rejection taking place. So this fits very well this paradigm, and we can also show that there is a massive destruction of donor cells in the native spleen, which is something that is expectable. So we contribute with this experimental model to this controversy, and which is still going on in the clinics because of this surgical advantages of doing splenectomy. But this is, from the immunological point of view, it's very risky, or you will increase the graft-versus-host disease risk doing the recipient splenectomy. And just to finish, I want to share with you some other example, and if this is possible, that this two-way alloreactive population can coexist in the clinics. So this is a clinical case which I will show. It's a pediatric patient that very early in her life has a ganglioblastoma resection with an intestinal location, which some years later finished in a portomesenteric thrombosis event due to complication of this resection in the midterm. And she received a multivisceral transplantation in April 21 with classic induction and immunosuppressive regime used in this situation. And she developed a graft versus host disease in the first month after transplantation, with a skin rash and a high percentage of donor-derived cells. So she received enhanced immunosuppressive treatment to control this graft versus host disease, and you can see here the drop in the chimeric proportion of cells in circulation. But unfortunately, some months later, she started with a post-transplant lymphoproliferative disorder. This is something that is a complication that is not infrequent in pediatric patients. It's associated to Epstein-Barr virus infection. So she has a primary infection of Epstein-Barr virus in these days that trigger this PTLD reaction. And here there is some of the clinical characterization of the PTLD. She after this PTLD diagnosis, she received different type of treatments with biologicals and to control and to kill the. This is mainly B cell proliferation. So she received anti-B cell treatment and antiviral treatment. And finally, she could control for some time the PTLD, but she entered in a very lymphopenic state. And in fact, the Epstein-Barr infection persisted and the PTLD was still a threat to her life. So the clinical team decided to just suspend the immunosuppressive treatment and to let the immune system clean the viral infection. And what happened is that she, being off immunosuppressive, she could control the Epstein-Barr infection, and she continued without any type of clinical signs of rejection and no more graft versus host disease for several months. Then after years, and this is an image of her intestine, the graft, either small bowel or colon, almost 3 years after being without immunosuppression. So this is one of the case of achieving operational tolerance in a multivisceral graft patient. That this is not very frequent. So the operational tolerance is defined as stable graft function and absence of rejection without any type of immunosuppressive treatment for at least 1 year. So this is fitting all this definition. So we want you to understand what happened that originate this operational tolerance state. So just looking at the lymphocyte count, this is the period of the lymphopenia and the between 100 and 200 days and the PTLD. And after releasing the immunosuppression, she recovered lymphocyte count in peripheral blood. She recovered all the different type of immune cell population in periphery, but without showing signs of alloreactivity or activation of either rejection or graft versus host disease. This seems not to be related with the number of regulatory T cells in periphery. She recovered the Treg population in periphery, but these levels are not very much similar to the ones that are seen in other type of transplanted patients, and either in counts or in proportion is the same situation. We also wanted to understand if there is some change in the type of cells that from donor or recipient origin in the graft. So finally, in this graft biopsies performed uh, almost 3 years after being released of immunosuppression, and also using this same technology or using a mismatch HLA antibody to see donor cells, we can observe that in lamina propria there are two populations of from hematopoietic origin, the recipient and donor cells in CD3 positive and negative compartment, and this is also confirmed by molecular biology STR analysis. And on the other side, at the structural cells, for example, the epithelial cells, there are most of them from donor origin. This means that there are still donor-derived cells in the epithelial compartment, and also in the intraepithelial lymphocyte compartment too, showing that there is a true chimerism in the graft. Another interesting observation that we have in this case is that the number of lamina propria regulatory cells is similar to the ones reported or observed in other patients, transplant patients. But what we observe is that even compared with the PBMC CD40 cells or to other transplanted patients, there is a much higher percentage of CTLA4 and also markers of proliferation. So T cells, this seems to be regulatory cells seem to be much more proliferative and more functionally more proficient than in other cases. And we have the opportunity to perform NGS analysis on the T cell compartment in biopsy samples by just looking at different biopsies performed at different types. So this representation shows the entire repertoire of BDJ, and the size of each square here shows the frequency of this, the appearance of each clone, and the colors are related to the type of sequence that are organized. So this is the picture at the before the engraftment and during the graft-versus-host disease. But what was striking for us is that after graft-versus-host disease and before just the withdrawal of the immunosuppression, there was a complete disappearance of any major clones, and the frequency of clones was much higher than later on. And this was confirmed by doing just comparison of the sequences, and we have 99% or almost 100% of new sequences arising after the graft-versus-host disease. And by also principal component analysis, we have these pictures of very different distribution of clones in the after the graft-versus-host disease, and also this change what happens 2 years later. So for us, this graft-versus-host disease episode has a dramatic impact in the renewal of these cells, and this may also be part of the origin of this operational tolerance. So we're still trying to learn more on this case, but this seems to be one of the key elements of the case. So as a final remarks, we can say that this two-way cellular-active paradigm can be also recapitulated in animal models, and this providing tools for developing new strategies for patient management. And in general, chimerism could be considered as a necessary condition for establishment of operational tolerance, and this has been shown also in other settings. But the removal of tissue-resident alloreactive cells, such as resetting immune system, can be also necessary. So still an open research field, but very interesting findings are arising in considering this two-way alloreactive paradigm. So finally, I want to ask people who work in my team and collaboration and did this work. Mainly the animal models have been developed by Pablo Stringa, researcher in my group, and the clinical studies were done by Dr. Rodrigo Papagovi in collaboration with the EDPAS team in Madrid, led by Francisco Hernandez, the transplant team, and Dr. Antonio Perez Martinez in the hematology group that was following the clinical case that I showed. So many thanks for your attention, and be happy to take questions.
Thank you so much, Dr. Rumbo. That was an incredible presentation. Um, uh, First, I mean, of course, anybody, if they had any question, you can write it on the chat and I can read it for you, or you can unmute and ask Dr. Rumbo, whatever you consider. First, I would like to ask you about the multivisceral transplantation model in rats. I mean, it's really hard to get an animal model on transplantation. I would like to ask you if you can share a little bit more about how challenging it was and how long it took to achieve that goal.
Yeah, well, this is really a hard work and is just take by my microsurgery team. So I would say normally to achieve this type of proficiency in microsurgery, you need to do a kind of training, a stepwise training. So we in general start by working in simpler models such as ischemia perfusion, and then some solid organ transplantation more simpler such as kidney, because the big thing here is the anastomosis. Just the vascular anastomosis are very, they are tiny, so you need to have a microsurgical equipment, microsurgical microscope, and skilled trained people doing that. And normally to have this training, it takes some years, and in our group, uh, we always start by doing a PhD and getting the training along the PhD, and then after some years, you are and working very hard, you may have these skills. I'm not doing that. I'm just discussing the immunology part with them, but normally they are microsurgical veterinary doctors that are doing that, and they are amazing. But you need to have a special part of your team dedicated full time to do this. This will be perhaps the take home message on this part.
Yeah, to be honest, not any group around the world got that
There are
kind of.
few, and there are few groups around the world doing this in rodents. Rats are perhaps a bit more challenging, less challenging, excuse me, than mice, and there are very few groups doing this in mice. Unfortunately, there is a huge difference between rat and mice in complexity, and we just have the rat models in our groups, and we are a bit limited on some tools to do more basic research on that. And we are eager to go for mice, but it's always challenging.
Yeah, definitely it is, but really congratulations, because that it's amazing to
Thank you so
see
much.
as you get that
It's
model.
for my
Yeah.
team. Thank you.
Can I ask a question, Alendra?
Yeah, sure. Please.
Dr. Martin, very impressive work, I must say. I just I want to ask one question, that the experiments that you performed in mice, do they have a certain kind of HLA haplotype? I mean, did you prefer some
Yeah.
genetic makeup of them?
Yes, in the sake of time, I could not comment, but we are using in these experiments Brown Norway as a recipient and Lewis strain as donor. In this sense, the normally is using Lewis derived graft. you always get more chance of having immune activation than in the other way around. We also have performed the opposite. And so if you to have a graft-versus-host disease, you need to have or you have more chance to have the graft-versus-host disease using Lewis as donor cells, but they have a mismatch in HLA class 2 in this model. But and it's not completely clear why this is not really symmetric. We have just performed very simple analysis, and we have observed that there is the CD8 population in the Lewis rat is a bit higher and more active, and we believe that this is why these grafts are more prone to do graft-versus-host disease than if you perform in the opposite way.
Thank you.
You're welcome.
I don't know if there is any other question from the audience. Please go ahead. You can write on the chat or unmute. Taking the question of Dr. Anis, I would like to ask you, thinking about translational science, because you show a clinical case. And I'm thinking what we can do from the clinic, because maybe now when we talk about transplantation and lab tests, we focus more on, like Dr. Anis mentioned, HLA matching, and also the presence of antibodies, autoantibodies, right? Cross-match by flow cytometry. So do you think, is there any other test that it could be, considered, especially in intestinal transplantation? Because for the model of what you mentioned, it looks like more, I don't know if I can say similar, but it looks like you had to take into consideration more features, like maybe in stem cell transplantation, bone marrow transplantation, more than on an organ solid transplantation, like a kidney, for example.
Yes, well, one of the things that we believe is that we are in a kind of intermediate situation in this type of continuum in the two-way alloreactive extremes. So in this type of graft in which you are just introducing organized lymphoid tissue with the graft, and you are introducing precursors, hematopoietic precursor with the graft, this is an intermediate situation. But just take into consideration that this in chimerism can be one of the drivers of this tolerance. One thing that is nowadays in clinical trials is the use of hematopoietic stem cell precursor transplantation at the same time, or just matching the solid organ transplantation from the same donor. So in order to try to get some stable chimerism and to achieve some operational tolerance state. But from the clinical point of view, mainly at the moment of the transplantation, the most important thing is to establish if there is a presence of previous alloreactive populations or antibodies at the time of transplant, as in the other solid organ transplantations.
Yeah, I see. Yeah, it's also, I mean, you got a gap on time because of ischemia, reperfusion injury. Absolutely. So I'm also thinking about that, because I guess it's not the same in a kidney or in a.
Absolutely.
Right?
Well, one thing that is coming in solid organ transplantation is all these perfusion machines that allow to condition the organ and not doing just static preservation, trying to minimize the ischemia reperfusion injury, but also you have there a window of opportunity to treat, to condition, to induce some anti-inflammatory state in the organ, and this is something that it's a very exciting field in cellular transplantation also.
Can you comment on the time set of the reversibility of this tolerance? And is there a mismatch between the recipient and the host? And it's a particular SNP on the CTL4. Did you explore this for having more Tregs? So it's just a comment, please.
Yeah, thank you so much. So related to HLA matching in the clinical case, there was a full mismatch in class 2 and DR and DQ, and also there was a mismatch in HLA A and B. So there is not a kind of compatibility that as a driving of the operational tolerance. It's something that we believe is related to just re-education of the two alloreactive population and enhancing some tolerogenic response. We believe that this graft-versus-host disease episode that really deleted most of the cells in the periphery in the graft, because the problem normally are the memory cells that are residing, tissue resident memory cells that are very difficult to stop with the immunosuppressive treatment. So this combination, we believe that is important, but it was just something that arose that we did not provoke, and we are trying to set up some experimental models to see if we can then manipulate this type of reactions in order to get the tolerance. Then two things that I can say is not really lack of response, because the patient received vaccines and could mount responses against third party and against also immunization programs. And another thing is that it's always a threat how stable this tolerance is. Normally, a kind of viral enteritis or something that an infection affecting the grafts may also reignite some allorecognition event. So this is always a very situation in which patients should be follow up very carefully, just to try to very quickly detect restarting of alloreactivity. But so far, the patient is stable and with operational tolerance.
Thank you. Thank you for sharing with us.
Thank you.
I don't know if there are any questions for Dr. Rumbo. Really, thank you so much for your presentation. Congratulations. It's an incredible work. Thank you for sharing your research and your knowledge with us. Thank you so much.
It was a pleasure.
No, please, it was our pleasure to have you here. So I will ask if you can wait just a little bit. Dr. Anis, she was online. She asked you a question, but she got some problems with the connection. She's trying to reconnect again. I mean, to join us on the Zoom. She just texted me, so if we can wait just 5 minutes for her, so she can join us again, and we follow with her presentation. So let's wait 5 minutes, please.
So I'm sorry, I was disconnected actually.
Yes, there you go. Yeah, I was saying. Yeah, I was just saying that it's okay, Dr. Anis. Thank you. So please let me introduce you to the audience.
Yeah, meanwhile, I'll share my talk.
Yeah, please. So thank you so much, Dr. Sabia Hanis, for joining us today. She has fellowship of the College of Physicians and Surgeons of Pakistan in immunology, MCPS in clinical pathology, and a diplomacy of the Royal College of Pathologists in immunology. She is a consultant immunologist and section head at the Indus Hospital and Health Network in Pakistan, She's also chair of the Continuing Medical Education Committee and the Quality Improvement Committee in the same institution. Previously, she worked at the Saint Institution of Urology and Transplantation as an associate professor of immunology and was instrumental in the development of the immunology department. She's a founder member of the Pakistan Society for Immunology, and she was General Secretary from 2012 to 2022. So thank you so much for joining us today, Dr. Anis. I'm sorry for my pronunciation of some of the institutions and names. Maybe I try to do my best. So really, it's really a pleasure to have you here because, as the others may know, this is a course on HLA and transplantation, remarking the highlights in transplant immunology, but also on HLA-related diseases. So that's why we got Dr. Anis here. Thanks again. And if you got any questions, please, you can write on the chat or unmute your mic and ask Dr. Anis whatever you consider after her presentation. Again, thanks, Dr. Anis, for joining us. We are so happy to have you here.
Thank you, Linda, for such an explicit introduction. Thank you very much. And thank you, the team of Immunopedia and Immunology Society of Argentina, for giving me this opportunity to interact with such a wide-scoped intellectuals on a global forum. So after Dr. Martin's such an impressive presentation, you'll find my presentation very basic, very simpler, and I hope that it will incite your the excitement and so let's see how we go from here. So I don't have anything to disclose. And then these are my objectives. I'll be discussing about the HLA molecules, their basic structures, the genetics, and the organization. And then the mechanism of antigen processing and presentation by HLA molecules to T cells, and why we call them as a molecular gatekeeper, and what are the association with other immunological disease or other diseases. And then we'll explore some applications in the diagnostics, therapeutics, and personalized medicine. And we'll look at some future perspectives. So human leukocyte antigens, also called as major histocompatibility complex, one and the same thing, is actually a cluster of gene complex encoding HLA molecules or MSC proteins. Actually, they were first discovered as MSC genes that were responsible for the acceptance or the rejection of transplanted organs in mice. So that's why they were actually named major histocompatibility complex. But then in 1954, Jean Dossett and Jan van Rood, they found out the similar kind of genes and expressed proteins on the leukocytes of the patients who had multiple pregnancies and blood transfusions, or they had received kidney transplants. So they named them as HLA or human leukocyte antigens. As you can see that all our cells, they have a lot of HLA molecules on our surface. So they are located in all the cell surfaces in our body. So if you look at the genetic composition or the classification of the HLA genes, they are actually divided into three classes: class 1, class 2, and class 3. The class 1 and class 2 HLA antigens, they are actually associated with antigen processing and presentation to the T cells, while HLA class 3 antigens, although they are called as HLA, but they are not associated with antigen processing or presentation. And you can see that they're actually associated with other immune responses, important immune responses. For example, the complement proteins, some cytokines, and heat shock proteins. And so the HLA class 1 molecules, they are actually found on all nucleated cells, whereas HLA class 2 molecules are found explicitly on antigen presenting cells. And antigen presenting cells, we know that basically they are dendritic cells, macrophages, or B cells, but in certain circumstances, some other cells may start expressing these HLA class 2 molecules. So a little bit of more detailing of the HLA class 1. The genes encode 3 major and 3 minor HLA class 1 proteins. The major HLA class 1 proteins, they are found on all nucleated cells, include HLA A, B, C, that present antigens to CD8 positive T cells, while minor HLA class 1 molecules include HLA E, F, and G, and they have their role in immune regulation, in pregnancy, and in some cancers. So we'll learn a bit more about these in details. And these are actually the diagram of the HLA class 1 molecules. As you can see here, this is actually the crystalloid form, and this is the schematic diagram of these HLA class 1 molecules. And as you can see that HLA class 1 molecules comprises of alpha chain, which has 3 domains then, alpha 1, alpha 2, and alpha 3. And there is a light chain which is called as beta-2 microglobulin. Now, if you look at over here, the crystalloid form, these are actually in the form of beta-plated sheets. And the genes of these HLA class 1 molecules, as we've seen, is on the chromosome 6, whereas the gene for the beta-2 microglobulin is found on the chromosome 15. So beta-2 microglobulins, when bind with major and minor gene subunits, it produces a heterodimer. And when the peptide gets bound on this peptide group, binding group, it becomes a heterotrimer. So the MSC class 1 molecules are heterotrimeric molecules. And in this form only they are stable and when they're expressed on the cell surface. And then there are other molecules like MIC-A and MIC-B, which are MSC class 1 like antigens A and B, which are not HLA molecules, and they have their other functions. Now, moving towards MSC class 2 molecules, the genes express 3 major and 2 minor MSC class 2 proteins, including HLA-DP, HLA-DQ, and HLA-DR. And each molecule again have alpha and beta chains that are encoded by their respective loci on the genes, which is on chromosome 6. So both of them, all of these, they have alpha chain and the beta chain. And these alpha, and as you can see in these diagrams also, again, actually class 2 molecules are also in the form of beta-plated sheets. And this is the schematic diagram showing the peptide group and found by the peptide binding group, which is formed by alpha 1 and beta 1, whereas the alpha 2 and beta 2, which is it is not that polymorphic, and then it is attached, goes through the transmembrane and is anchored into the cytoplasm. Again, there is a peptide in the peptide binding group, and this makes it heterotrimeric, which is then expressed on the cell surface to be presenting antigens to the CD4 positive T cells. So the proteins of class 2 combine to form heterodimeric alpha by the combination of alpha and beta chains. And then when they're expressed on the CD4 positive T cells, they are actually heterotrimeric. Then there are other minor MSC class 2 molecules, including HLA-DM and HLA-DO, which have functions in HLA processing and presentation that we'll see when we'll go a little bit in details. So HLA molecules are very important in T cell immune response. They're essential for T cell mediated adaptive immunity. The T cell recognizes foreign antigens produced by the intracellular proteins, which are processed and presented by HLA class 1 or class 2 HLA molecules. As you can see over here, these are being presented by HLA class 1 or 2 molecules to the TCR. So on recognition of HLA-associated peptides by TCR, the several T cell receptor proteins, as you can see over here, they come and they are recruited to this side, making this interaction between the T cells and the cells presenting the antigen in the association with HLA molecules very strong and strengthen this interaction. And so the cell activation occurs because of the signal transduction going into the T cells, and then the T cells are activated to perform their various functions, For example, CD8 T cells have a cytotoxic function. They kill the infected and the cancerous cells, whereas CD4 T cells have a wider range of effector functions, like they are helper T cells, though they can be either Th1, Th2, Th17, Th9, Treg, and others. So if someone who do not have HLA molecules, you can imagine what can happen, because it will, of course, it will lead to immunodeficiency, which is a kind of immunodeficiency because of the HLA molecules, because these are very important in our immune response, T cell mediated immune response. Now look at the HLA class 1 processing and presentation pathway. The class 1 pathway is actually for the processing of intracellular antigens. These HLAs actually, they also guide the type of immune response as related to intracellular organisms or antigens, or it is extracellular antigens or organisms. So to the processing, for the processing of the intracellular antigens, when there are any virus is taken up, it is then endocytose, and then its proteins are produced into the nucleus or into the cytoplasm, and or the defective proteins that are produced in the cells. They all are actually processed through this pathway. First, they are ubiquinated, and then they go ubiquinated means they are actually tagged with this ubiquitin proteins, and then when they pass through the proteasome, they have proteins for proteasomal degradation, LMP1 and 2, which chop off these proteins into small peptides or bigger peptides into smaller peptides. These then transported to the endoplasmic reticulum with the help of transporter associated with antigen processing into the endoplasmic reticulum. Now, the MSC class 1 proteins, they are being actually synthesized into the ribosomes. And from there, they are translocated or located into the endoplasmic reticulum, into the lumen. And once they are into the lumen, they get assembled, and the proteins, the peptides are coming here through the tap, the HLA molecules are coming here, and then there are some chaperone proteins like capucin, reticulin, calexin. They and then ERP57, they form a complex which helps in loading these peptides onto the assembled HLA class 1 molecules into the endoplasmic reticulum. Once this peptide sits into the peptide group, now it becomes a heterotrimeric molecule, and now it is stable to move on. So this comes out of from the endoplasmic reticulum, passes through the Golgi apparatus, and then it expresses on the plasma membrane to be recognized by the CD8 positive T cells. Now, HLA class 2 processing and presentation a little bit different, because it is for the processing of the extra cellular antigens, and extracellular antigens are actually taken up by the cell by phagocytosis mostly. And once they are phagocytosed, they are either in the lysosomes or in the endosomes, and their disintegration or degradation occurs into the smaller peptides. And these smaller peptides, then they move into the vesicles, also called as MSC class 2 compartment, where they are going to bind to the HLA class 2 molecules. Now, from where these HLA class 2 molecules have come into these vesicles, they again are first synthesized into the ribosomes, and from there they are translocated into the endoplasmic reticulum lumen, where they get assembled. And when they're assembled, there is an invariant chain that gets into get fitted into this peptide binding group. This is important because only then it becomes a heterotrimeric molecule to be stable enough to move out of the cells for the rest of the processing. And if it's not there, then other irrelevant antigens are going to sit over there here, making it unuseful, non-useful, or useless. So these, along with the invariant chain, it passes through the Golgi apparatus, and then it goes into the MSC class 2 compartment, where we have HLA class DM and some tissues, HLA class DO molecules. What they do is They chop off this invariant chain, and then the remaining portion is called as CLIP, class 2 associated invariant peptide. Then this HLA-DM molecules, it causes it removes this clip from the peptide group, and then it locates or makes the peptide that is present over there, sits on this peptide binding group. Now it becomes a stable heterotrimeric HLA class 2 molecules, which is then comes out of the cells and get expressed on the cell surface and be recognized by the CD4 T cells. Now, there's a process called cross-priming also. What happens in this? In some cases, some of the antigens, they have to be presented to both the CD4 and the CD8 cells. For example, some cancerous cells, some viruses. Why? Because for the naive CD8 positive T cells, they have to have the help from the CD4 T cells to make them more effective. So, so the antigens, when they are actually processed, and by both the pathways, class 1 and class 2, they are going to be presented to both CD4 and CD8 positive T cells. And CD4 positive T cells actually help out CD8 T cells because they are not activated for the same kind of antigens to make them more effective to produce immune response against those foreign antigens or viral antigens or the cancerous antigens. So this is this is called as cross priming. Sometimes, some of the peptides, they actually leak or travel to the adjacent cells also, and over there they can again be processed and presented by the antigens over system over there, and that is also called a kind of cross priming. Now, there are two important characteristics of the HLA molecules. One is that they are codominant, and other is hyperpolymorphism. So let's see how what does it means. Codominant, as you know, as the blood group, the alleles of the blood groups, they're codominantly expressed. So if someone has a blood group A, the both of the alleles are AA, and someone who have the A and B alleles, it will be expressed in A and B. So similarly, in these HLA molecules, we have both alleles. One is from the father and one is from the mother, and they are codominantly expressed. Both the alleles are going to be expressed on the cell surface. So in this diagram, you can see that A has 1 and 9, B has 2 and 5, and HLA-DR has 3 and 6. So it will make HLA-A1 9, B2 5, and DR 3 6. So this is actually called as haplotype. A little bit about the HLA inheritance. It actually follows the Mendelian law of inheritance. So there is actually, if looking into the offspring, there will be 50% chance that there will have one haplotype match, and 25% chance that there will be not identical, and 25% chance that there will be no match. But what is important is that each of the offspring will have one haplotype match with a father and one haplotype match with the mother. Any offspring you can locate over here, you can see that there's one haplotype has come from the father and one haplotype has come from the mother. So this is a very important thing that we actually use it to check the paternity in the HLA lab when we are doing live-readed transplants. So when to look at the ambiguity, if there is over there, or if the patient's relatives or the patient's guardians who have come, they are actually the parents or not, are not, and vice versa. So you can see that there are a number of alleles for each genes, and then these alleles are expressed in the form of proteins, but not all the alleles are expressing HLA proteins. Some of them are null alleles. So if you can see over here for the HLA-A, there are more than 7,000 alleles, but the proteins that are expressed are 4,355. But look, what is important is over here is by looking at these numbers is you can appreciate that the HLA has a very high polymorphism. There are a lot of HLA molecules that are expressed in a population, and also in single individual. There are so many HLA molecules that are expressed on the cell surfaces. Then there is the phenomena called linkage disequilibrium. What happens in the linkage disequilibrium is, as you can see over here, that some of the haplotypes, because the haplotypes, they are actually inherited in block. So the in linkage disequilibrium, certain HLA alleles that are inherited together more frequently than predicted by random assortment. So, for example, in a population, in a big population of several lakhs, they may have A3, B7, D, R3 very more commonly occurring as compared to other haplotypes occurring in association with each other. And this has implication in the transplantation. Now, the first of the HLA gene that was sequenced, it was in 1998, or this, and it was then the data was actually released for which has this collection of all HLA genes or HLA sequences. It was initially called as IMGT HLA. International Immunogenetics genetics data, and then later on it was termed as immunopolymorphism database. So the latest release was in 2024, and then they have been showing around more than 25,000 alleles, which had been sequenced for 45 genes, which are located within the MSC molecules of the human genome. So if you want to see what are the latest updates, you can always go to this site and you can look at this site, what are the new genes that or the new alleles that have been discovered now. So looking at this graph, you can very well appreciate that starting from this point, 1987, then when the 1998, when this data was released, now we have an enormous numbers of these HLA alleles contributed to this database base. Now, we have seen that these HLA molecules are very highly polymorphic. So why? What is the reason? This high polymorphism of the HLA molecules actually translate into the response to infections, to the responses to the allergens, the injury, even the response to the vaccination that differs so much between the individuals. So this variation in the responses to different injuries or allergens or infections is actually because of the different types of the HLA. So why there is a need for this polymorphism? It is very needed. It is actually for the survival for the fittest phenomena, because it ensures that at least some individuals within a population will be able to recognize protein antigens produced by virtually any microbe, reducing the likelihood of invasion by a pathogen that may evade the immune system of all individuals or by mutating their antigenic peptides in a given species. So it is actually the question for our human survival. If all of us had same kind of HLA type, and if some pathogen would have overcome our HLA makeup, then this human race could have been extinct now. So this is actually because for our survival that there is high polymorphism in the HLA molecules. So, so many molecules which are inherited, both the alleles HLA from the mother and the father, they are expressed on the cell surfaces, and then they are presented to the CD4 and CD8 positive T cells. Now, what is important is the peptides which are being recognized by the TCR, the epitopes, this is or the molecules, they are different from which are actually anchored into the this peptide binding groove. They are different, and also these MSC molecules, they do not distinguish between self and non-self. So where this property comes, it actually the T cells, they are very efficient in discriminating between self and the non-self, and also they recognize antigens only that are bound to the self MSC molecules. So from there, the specificity comes. HLA molecules are going to display all kinds of antigens, whether it be self or non-self, and most of the antigens that are displayed by these HLA molecules are actually self molecules, and the non-self molecules or from the foreign antigens are actually rare compared to self molecules. So the role of human leukocyte antigen, what is it? Then in our for our survival, it is actually a gatekeeper molecules. It actually protects us from all the invading organisms, and it is also our identity card, because if each and every one of us have a different kind of different set of HLA makeup, so it actually is a molecular ID card for us. And also, it is important, of course, to ensure the resistance against the infection at the population level, in fact. Also, it is important in the organ and bone marrow transplantation. Dr. Martin has very explicitly and very nicely explained all this in detail. And then besides these major HLA molecules, HLA-G molecules and HLA-E molecules, now we have come to know that their important role during the pregnancy, because of this immunotolerance effect. Because the fetus is actually foreign to the pregnant woman, but then why it's surviving? Because of effects of the HLA-G molecules, because they actually suppress the maternal NK cells from invading the placenta and the fetus. So for every infection or any vaccine or meal, that are all influenced by our HLA makeup, HLA type, and that they actually determine how our body is going to react, or we are going to react to different antigens. Now, they are also very important for the tolerance against autoimmunity, because the T cells, they train the T cells to help the autoimmunity. So we know that these T cells are developed in the thymus, and over there actually class 1 and class 2 molecules are expressed. So the T cells, they are going to recognize the antigens over there in association with actually class 1 and class 2 molecules to be for their survival or maturation. But those T cells that they recognized these molecules with high affinity, they die over there because they are their autoreactive. So they actually die over there in the thymus only, and or they become TREG cells. In the periphery, if you look at this diagram, the cells that express these MSC self-antigens in association with MSC class 2 molecules, especially because they are more associated with autoimmune diseases, they are very rare as compared to the other antigens or the foreign antigens. So these are actually the ways how these HLA molecules train T cells to prevent autoimmunity in us. So if anything goes wrong in the system, of course, we'll have autoimmunity or autoimmunity. So there's so many studies available. They're showing association of different diseases with different HLA molecules, but the ones which are actually very strongly associated and they're actually used in the sometimes in the diagnostic workup are HLA B27 and HLA DQ2 and DQ8. So HLA B27 associated with ankylosing spondylitis, and HLA DQ2 and DQ8 associated with celiac disease. And sometimes, not in all cases, but most of the cases, on some cases, we have to use this HLA typing to come to a diagnosis of either ankylosing spondylitis or celiac disease. Now, there's so many studies available who have looked at the association of different HLA molecules with different pathogenic diseases. For example, recently in the COVID infection, there was a lot of people have published the association of HLA molecules with COVID-19 or the response to vaccination to the COVID-19. And a lot of studies have looked at the association of these HLA molecules with the TB and some other leprosy and some other infectious diseases. But then these needs to be taken with a pinch of salt, because how to prove that this association isn't real? Because we actually did a study looking at the association of HLA molecules in SC positive positive patients who were making cryoglobulins or having cryoglobulinemia. And before that, the published studies have shown the association of this phenomena with the HLA class A2, BA, DR3. And these actually haplotypes, this haplotype or these antigens are very common in our population. But when we did the study, we found a different set of sets of HLA molecules that were associated with cryoglobulin formation in our SCV positive patients, that were HLA B57 actually, as far as I remember, actually, and then the A2 was there. So what I want to emphasize is that even this association of these HLA molecules with different studies may differ in different kinds of population. The strong association like HLA B57, B27 with ankylosing spondylitis is also not at the genetic levels, at the allelic or suballelic levels. So there was a study in which this shown that some of the alleles, they're actually preventive against the ankylosing spondylitis, whereas some of the alleles, they were actually associated with ankylosing spondylitis. Again, in celiac disease, actually, it is actually DQ.2.2, and it has been proven that why it is associated with celiac disease. So all of these needs actually a lot of experimentation and studies to actually prove these associations. Now, for infectious disease susceptibility, the actually B57, we know that it has protection against HIV progression, whereas B35 may increase the risk of HIV infection. So, how this association can be rationalized? They said that efficiency of antigen presentation affects how quickly and effectively pathogens are cleared. So, some actually molecules may process and present these antigens to the T cells very effectively and very quickly, so that the pathogens can be cleared effectively in time. And while the others may not do so. So that may be the cause behind this phenomena. Also, there are some HLA molecules linked to the adverse drug effects, like HLA B57 is a well-known cause of hypersensitivity to abacavir, which is an HIV anti-HIV drug, or HLA B15 is associated with severe reactions like Stevens-Johnson's. So what is the mechanism? Could be over here. It says that the drugs which are when processed and presented in association with these HLA molecules, they actually form new antigens, and these new antigens actually causes inflammation, trigger inflammation and causing adverse drug reactions. So what could be the implication of this HLA linkage with the drug adverse reaction? So it can be in the personalized medicine. We can do the HLA testing of the patients in which we are going to institute these drugs, so as to see if they are exposed to the side effects or not. Now, HLA typing is done in the labs. Initially, it was only the serological typing was done, in which the B cells and the T cells were taken out from the peripheral blood, and then they were incubated with the antibodies, anti-HLA antibodies in the plates. The HLA specific to the antibodies makes the complexes on these cells, and when we put the complements, the reagents, the antigen antibody complexes, they activate the complement pathways, and then those cells which have those HLA antigens reciprocal to the antibodies in those wells are actually they die, and we can see them under the microscope either by staining them with the eosine or by looking at the fluorescent dye inside it. And this is how their reports are then generated and reported. Then came the polymerase chain reactions assays, in which there were sequence-specific primers. This is also a low resolution HLA typing as was serology, but more sensitive, more specific. Then came the sequence-specific oligonucleotide primers based, and then now we have NGS methods. So which are now we have a lot of data that has come out because of the NGS, and the reporting has also been changed. And with the NGS, we look at the reporting of the at the 4-digit. Sorry. So this is how we get the reports when we are doing the high resolution HLA typing. We have HLA, which is the prefix, then we have a gene, then we have field 1, which is separated from the gene by the anesthetic. And this is from and up till this, it is actually coincides with the serological typing. And then we have the specific HLA protein. So in this case, it will be actually HLA 02101. And then it is then the other 01 is the one which shows which is synonymous DNA substitution, and then there is a non-coding region, a suffix with the suffix used to denote the changes in the expression. Uh, so what is the utility of HLA typing other than bone marrow stem cell transplantation and solid organ transplantation is we know that sometimes we have to go for the HLA typing like HLA B57 or HLA DQ2 and DQ8 for making a diagnosis. In fact, HLA B51 is also used, although it does not have a very good sensitivity, but it is used to diagnose Behçet's disease. Then the risk for developing a drug reaction like HLA B57.02 associated with side effects with the abacavir, we have to do the patient testing to see if it will be beneficial or it will be risky to giving this drug. So now what's new? What's more exciting than in HLA world? MSC tetramers. These are actually the laboratory tools. These are the reagents by which we can monitor or quantitate the T cell responses to specific antigens. So these are made in the lab. You can see over here four HLA molecules with the specific antigens. They are bound by they are biotinylated, and when streptobidine is poured, they become a tetramer, and then it is conjugated with a p fluorochrome. And when it is reacted with the antigen-specific T cells, it can be enumerated or can be tagged or can be seen on the flow cytometer. As you can see over here, these are the CD8 tetramer. So you can actually quantitate these T cell responses by this test. So its application is in finding the allergen and the epitope identification in allergies or infectious diseases, or autoimmune diseases, tumor immunology, or for protein therapeutics. They are also used to judge or gauge immunotherapy efficacy, immunomonitoring, to look at the T cell mechanism, to look at the vaccine efficiency, and so on and so forth. So it has a lot of application. Maybe Dr. Martin was in the presentation by Dr. Martin, you can use these kind of tetramers to look at the responses in his experiments. So role of HLA-G in tumor immunotherapy is now the hot topic for the research. We know this HLA-G is an immunotolerant HLA molecule, which is associated with the propagation of the pregnancy or a successful pregnancy. But if it is present in the tumor environment, because of its immunosuppressive effect, it is going to lead to the escape of these tumor cells from the immune system surveillance. So so there is a need that we should look at this at the molecules that can block this HLA-G molecules ILT interaction that can restore host anti-immune tumor immune response, providing a strong rationale and opportunities to develop this targeted immune checkpoints therapy. Now, there's very interesting research I came across when I was preparing this presentation is the influence of HLA on human partnership and sexual satisfaction. So actually they enrolled 508 patients, and they gave them a questionnaire to answer those questionnaire, and they did the HLA typing of those individuals or the participants. And what they found interestingly is that this similarity in the HLA molecules is actually associated with a good partnership or a happy ending. So they concluded that actually mediates mate behavior in humans. So the young participants over here may think about it, getting their HLA typing done in them and in their partners to look at what type of life they will be conducting. So to summarize, our HLA molecules have a significant role in our daily life, and they're silently deciding how we live, how we heal, and how we survive. So they have a lot of influence on us. in our daily life. And actually, polymorphism, it shapes our immune system, how we handle the infections or respond to drugs. And so it helps in personalizing our immunity. Personalized to help in the personalized medicine, to help personalized response to infection or oriented chemotherapy and so on. Certain HLA variants influence outcomes of the infections like HLA-B57, and some of the HLA variants, they were associated with severe COVID-19 or malaria infections, but there's need to do more researches and experiments to prove these things. And also the interaction with medication and association with the drugs, it also we need to look for other associations so that we can have a personalized medicine for drug therapy. Now, what's on the horizon now? What should we be doing? We believe we should actually look at the tetramers. We should increase its utility in our experiments. We should be looking at the anti-tumor therapy, utilizing the HLA, the role of HLA molecules, the role of HLA in personalized precision medicine. We should explore it. And then, of course, it has a utility in developing a biorepository for umbilical cord stem cell. It should be global. It should be with the cooperation of global. It should be a global cooperation to help our generation and to help our survival. And of course, it's affecting our daily lifestyle, as we have seen. So, we may fail in our research sometimes. We get upset, but there is no need, because this is actually the first step towards our success, I believe. Thank you very much for your kind attention. And if any question, I'll be happy to take. And I can, this is actually I've prepared actually a quiz. If you are interested, you can, attempt that. I will put it into the chat box. Thank you.
Thank you so much, Dr. Anis. Yeah, please, if you can share the link of the quiz on the chat, so the audience can get it. Yeah. I think it will be better if you stop sharing the screen. Maybe that may help you. There you go.
I hope everyone can see this, and if anyone has any questions, please let me know.
There, I was muted. So yeah, it works. I'm actually checking the quiz, so we can, if you agree, we can give 5 minutes so people, the audience, can check it, and then we go for the questions.
Sure.
Yeah. If someone else got any question, please, you can unmute or write it on the chat. So let's check for the quiz for 5 minutes. Also checking the question on the quiz. If everybody has any questions about that, you can also share questions or any suggestion you guys may have. Dr. Anis, I would like to ask you, well, first of all, thank you so much for your presentation. It was so complete. It actually gets from another point of view of HLA testing. Maybe when we think about HLA typing, we are more focused on transplantation and histocompatibility, but there's so much more involved, not only in autoimmune diseases, but also infections and drug hypersensibility. You mentioned about abacavir and HIV. Actually, when a physician wants to prescribe abacavir in HIV or people living with HIV, it's a mandatory testing because of the serious consequences it may have for an hypersensitivity reaction.
Yes.
So I would like to ask you how you manage that in your lab. It's something that you got requested. I mean, in all of this points of views of HLA testing, it's for autoimmune diseases, it's for drug resistance. How is the statistics in your lab about it?
Okay, so for the HLA B27, we are doing for ankylosing spondylitis in a lab, and then the HLA DQ2 testing for celiac disease is very seldomly required over here. Then we would go for that. And then for abacavir, this is actually the domain of the ID specialist over here. And then if they think they have to give, and they want to do this HLA typing, they request it. So these are actually the tests that we are doing for the autoimmune disease, because other than that, there's not very proven, things that we have to do this HLA typing for all the autoimmune patients. It is not required over there. There are only some patients. Yeah, like actually B51, yes, we have done for Behçet's, when we're suspecting Behçet's disease, and so on and so forth.
Yeah, thank you very much. We have a question for the audience from Sylvia. She says, very nice presentation. Is there evidence of a specific HLA haplotype associated with susceptibility or protection against long COVID?
Yes, so there's so many studies. I mean, I've gone through that. Actually, the groups are actually looking at these associations, but there's, as I said, that if you find so many associations, but at this time point, I don't believe that these are actually in real. We have to do a lot of things, I mean, there are so many co-variables that are attached to that we have to actually take out those co-variables, covariates, and then we have to look at the association in reality. So at this time point, there's no certainty, but yes, studies are being done, and let's see if we have a consensus on it or not. And as I said, that it may also differ according to the population. As when we did a study in cryoglobin positive patients, we had a different kind of HLA molecules we found who had this cryoglobin positive and SE positive patients. But yeah, of course, but that may be because of the by chance. We have not proven it. Yes, it's for certain that these are actually this is actually the limitation of the studies right now. So maybe using the tetramers that I have shown you, with the help of those and the HLA molecules and specific tetramers we can use, and then we can look at those specific immune responses, and maybe able to prove something out of it.
I see. Yeah. Well, Celia, here she's thank you very much for the answer. If you have any other question, please, you can again unmute yourself, ask a question, suggestion, or write on the chat. I would like to ask you, it's something we try to do here in Argentina because these are genetic testing, the viability around the world is different, and also associations with diseases or infections. So maybe because we use international reagents, I mean, the reagents we use for HLA typing are usually from Europe or United States, where they use another kind of databases because populations are different. We are different on diversity. So
Exactly.
we are trying to implement some kind of database or a genetic bank in Argentina about the most spread HLA genes in our country. I don't know if you're working the same way on Pakistan, if you're trying to implement a specific database from your country or a genetic bank, if there are any you work more
Right. Okay. Yeah.
regional level. Yeah.
Yeah. Right now, I'm not working on it, but because I'm not in the transplant center at present, but where I used to be in SIUT, they are doing a lot of this HLA typing over there, and they are actually collecting this database, and they are actually have some kind of collaboration also where they send this all these, some kind of what is it called as? I mean, they have some kind of collaboration also. So it's very important to have our own database, of course, and we have different kind of centers over here in Pakistan, not in SIUT, but there's some in Islamabad, some in Lahore. There are other cities where this data is being generated. So once we have all the data available, we may have a good repository or a good database over here, and we can, be able to look at the different kind of associations. And I'm sure that the HLA type or HLA haplotypes and the disease associations or the outcome of the any transplant or infections be different in different kinds of population. It has to be. And that is actually what is about the HLA polymorphism, right?
Yeah, exactly. And that's why it's challenging, because it's
It
not
is
the
very
same. Maybe
challenging.
associations in some population with a
Exactly.
specific infection or disease is not the
Exactly.
same in other populations.
Yeah.
So that's why it's
Exactly.
really hard to.
Yeah, I 100% agree with.
Yeah, that's why it's hard to implement sometimes. Maybe it's not like much for, I don't know, abacavir and HLA-B57, because that's really well.
Yes. So, when we know that what is actually the mechanism behind that
Exactly. Yeah.
mechanism, then we are certain that, like for celiac disease, we know how it's presenting the antigens, the gliadin peptides to cause celiac disease. So we know that there's a strong association. But when we do not know the right mechanism behind, then we cannot say with certainty that yes, this is actually the real association.
Yeah. I think that's
A lot of
more
work is
challenging.
required. Yeah.
Yeah. And well, the very last question I got for you, you mentioned about the MHC tetramers. And I think that's really, I think that's really important tool, because maybe we don't see the same correlations between different techniques. I don't know, for example, when we analyze allo antibodies or antibodies against a specific pathogen, and then when we go to flow cytometry, we don't see that correlation between. It also happens in transplantation for sure, but we don't see the correlation with the levels of antibodies and the results on flow
Because we're not looking
cytometry.
at the specific antigen,
Exactly.
specific antigen. So this may be an area to explore, because it's been in a while in the in the pipeline, this MSC tetramers. But I've seen that there are not many papers, or that it's not being studied. Otherwise it has a lot of utility in not only in the transplant, but in allergies and infections and other kind of things that we can actually look at the T cell responses or enumerate them, quantitatively with this technique. That may
Yeah,
be.
I think this will be a really interesting or really important tool for complete that gap, right? Because that
Yes.
way, with the tremors, we can study the
I'm
specific
happy you took this
T cells,
message.
right? We
Yeah.
can see the specific T cells with that specific HLA gene. That's
Yes.
really interesting. Yeah, so thank you so much for sharing, again, your knowledge. It was a perfect and complete presentation. Really, thank you so much, Dr. Anis, for being here.
Thank you for inviting me.
No, please, thank you for sharing with us and taking the time to doing this and the quiz. That was a really complete quiz. Thank you, really. So if you got any questions there, we can move on with our next speaker. Okay, yeah, also the audience is really happy about the presentation. And we got so many messages on the chat. Thank you, Dr. Anis. So let's move on with our next speaker, Dr. Luis Cristobal Porto. Thank you so much for joining us today, Dr. Porto. So let me introduce yourself for the audience. Dr. Luis Cristobal Porto, he's graduated in medicine from Rio de Janeiro State University in Brazil and received a doctorate in human biology from Claude Bernard Lyon First University in France. He's a full professor and coordinator of the histocompatibility and cryopreservation laboratory at the Technological Core for Tissue Repair and Histocompatibility, and coordinator of the master's programs in health, laboratory medicine, and forensic technology at Rio de Janeiro State University. So, Dr. Luis Cristobal Porto, thank you so much for joining us today. It's really a pleasure to have you here.
Thank you very much, Alejandra. It's a pleasure to be here and to be with you and your audience. And thank you for the previous presentations that introduced well the thing, the subject, and I'm very glad to be here. Are you seeing my screen? Yeah.
Yes, we can see. Yeah, we can see now the full presentation. Thank you.
Okay, so I'm involved with a Brazilian group that is studying type 1 diabetes. So previous presentations introduced that some diseases and some autoimmune diseases are linked to HLA. So one of these diseases are well noted by Dr. Anis, that class 2 alleles are associated with type 1 diabetes as one autoimmune diseases. This is a large group that started to study diabetes type 1, and type 1 diabetes, it's autoimmune diseases, and there is a destruction of the beta cells in the pancreas. So these individuals do not produce insulin, so they have a very bad control of their glycemic level, and it's associated with morbidity, mortality, and there is a decline in life expectancy. So moreover than that, there is a budget finance to associate with the health ministry. So this group started to look for understanding once from 2008 to 2010 for the demographic clinic at the Brazil level. So at that moment, 3,600 individuals from different cities in Brazil were screened, interviewed, and they resulted in some information to the health ministry. There is another wave that is conducted by Professor Marília Brito Gomes that also works at my university, And he was very interested in the genomic aspect, as we have some information already that this association with HLA. And also at the university, we have one lab that was really interested in ancestry. So we came together to provide a deeper insight into pathophysiology and complications with these individuals that are associated. So these participants had are 10 years old when they are first diagnosis of diabetes. They already had 31 years old, and they're more women than men. In the first wave, 56, and the second wave, about 56 again. So this first wave had conducted that it's very difficult to those people control their glycemic levels. So their HbA1c, it's persistently high. So this is a major problem to control the complications of the diabetes. They deal with the difficulties sometimes to have insulin, and they are changing for the different teams to achieve targets to control those the life of those patients. And there is problems with the insulin pumps in different sets. So there is a big information for the health ministry to how to control and to have better health care to those peoples. So the problem, one of the major problems of diabetes, there is complications with diseases in the kidneys, diseases in the nervous systems, with different proportions. Sometimes people have both more than one different complications, and also controls of the glycemic states. But the major integrating question for Professor Marília Brito Gomes and the group, that it's an increase of type 1 diabetes from 1986. We have 10.4 cases per 100,000 people, and then the same number in 2006. But in 2015, we noted that there is an increase to 12.8 per 100,000 people. So, sorry. So the other thing that is a difference on the life expectancy and treatment, if they are treated in better conditions, they have better life experience, they have less complications, and this is a major problem for regulating health characteristic in the Brazilian system that is very large. So a major question for intriguing this question, that is diabetes was considered as once a white problem people, but we now see that that's not only for Caucasian. There is a big relative proportion of non-Caucasian, and for a country like Brazil that have much more admixed population, it's very complicated to know we're gonna have only white people in the consultancy rooms that have changed a lot since 1980s. So, as Dr. Anis had said to us, there is an ethnic background for each population that has a different HLA background. So and now we say that more than 30 to 45% of the diabetes patients, both in Brazil and USA, are no longer white. So we go to the formation of the Brazilian society, and so we get back to these roots of settlement in America. So as we know that men started or first was detected in Africa and then go to Europe and Middle West and then go to Asia, and one group colonized get down to America, North America, and so from North America they came to South America, and there is also a possibility that from Polynesia they formed the citizens of South America. But we had a different movement after Europe came to South America, and from the Amerindian that are already in South America, we had migration from people from Europe, first Portuguese when the navigation started, and then also started French and Netherlands, Holland people came to make cities in Brazil. And after we had a big slavery traffic that came from Africa, and most of the population from South America in Brazil was from sub-Saharan Africans. We also in Brazil we have different migrations from other countries, from Italy and from Japan, and again a new wave from Portugal and Spain, and also inside South America more recently from Bolivia and from Africa, again with a big commerce with Angola, and also in the first war, between the first and the second war, from Germany and East Europe, and also from the Midwest. So there is a lot of people that came to Brazil and South America, and so we have a big confluence, and differently from other countries, Brazil has a big information on admixed people that marriage themselves, and this is fully understand by the Brazilian Institute of Geography that every 3 and 4 years make a census, and now and when we have to declare ourselves, it's a auto declaration if we are black, white, pardo, that is admixed, or from Orient, or most and from Amerindian or indigenous. So Dr. Brito Gomes has this chance to coordinate this massive, with more than 1,700 patients from different centers in Brazil, from the north in Belém do Pará, from Ceará in the northeast, and also in Bahia, Salvador, and in the central area from the Distrito Federal, Brasilia, in the south from Paraná and Rio Grande do Sul, with Curitiba and Porto Alegre, the capitals of these states, and also from the southeast, from Rio de Janeiro. We have two centers from the two principal universities in Rio, and also many centers in São Paulo, also in the capital, and in large cities like Bauru and Campinas. The first study that we have done was most related to this background genetic with indels, and at that moment Professor Daisy Silva had established with her colleagues an indel that could map Amerindian ancestry, sub-Saharan ancestry, and also Europe ancestry. With this 46 indels panel, she was able to look, and we see in this map down in the bottom here, in red and green and in blue, the different proportion of these ancestry in each line, each point, it's an individual. So there is a distribution of, and we see that many people had a strong percentage of every of this ancestry. So she had the opportunity to compare with different studies that had made also in Brazil with about 1,000 individuals. So at that moment, we compare those patients with diabetes to see if they have a different ancestry background, and also a different background as from their auto-declaration. And the difference was already know that we have different proportions of auto-declaration in the northeast compared to the south and southeast. There is a majority of white people declaration in the south, and there is a much more black people in Bahia, Salvador, and also there is a very large number of admixed people in São Paulo capital, Rio de Janeiro capital, and in the northeast. So there are differences in this ancestry background genomic, and there is an association with type 1 diabetes with a more pronounced background of European ancestry. But auto-declaration was have a good information, but as you can see, people that declare themselves as black, they also had some Europe background. And if they are admixed, they have more white Europe ancestry, and also have some black sub-Saharan African ancestry. There is very little from this type 1 diabetes patients with Amerindian background. As a second role for those students for complications, Dr. Marília Gomes, that is in the clinical set, also involved some PhD students to look for complications. One, the students is followed those patients with the information of nephropathy. We know that kidney disease is more predominant in sub-Saharan ancestry. we found some correlation, but as we have to do bone Ferroni correction, it was not possible to be so affirmative in this association with the African ancestry and kidney disease for those patients with type 1 diabetes. Another disease is the eye diseases. Patients with type 1 diabetes develop diabetic retinopathy that evolves sometimes to blindness, and it's very complicated. And so you can see the three first panels here are from the control groups, and then we have patients with the mixing of indigenous, European, and Black sub-Saharan ancestry. So there is also a very complex situation of admixture in this population, and again, we are not able to ascertain that is associated with genetic ancestry. So one of her students studied the HLA from a small group that has this diabetic retinopathy, and it was the first one study that included HLA, and for those patients, we can see that there are differences for the clinician set, but we saw that there are some alleles that are on the association with this diabetic retinopathy, and again, both we only studied class 2 alleles, so 1302 and 0301 had a very high odds ratio, but also again with the number that we have studied after bone Ferroni correction, we are not able to set this significance to a level that was confirmed and to generalize it. So we also explore haplotype, as Dr. Anis said, that is very common that all the HLA DRB1, DKA1, and DKB1 came together from a father and the other one from the mother. So we look that probably one of these haplotypes could be associated to an ancestry. And again, we found again DRB1 0401 that is already known that are associated with diabetes, and 0301 and 0302 for DKB1. So this is a very common haplotype that is observed already in Europeans and in United States, and it was very also present in the Brazilian population. But for those people with diabetic retinopathy, we found 1302, 0102, and 0604. But again, I should emphasize that bone Ferroni correction did not confirm this difference. We had the opportunity to work with Dr. Azulay, and another center enter this cohort, and this is in Maranhão state. It's São Luís, the capital of this state in northwest. And at that moment, we had the opportunity to look as he had at the same center controls patients that are excluded to have diabetes type 1, and they are controlled, and also the diabetic type 1 patients. So they are investigated at that time not only for HLA in the group of patients, but also for genomic ancestry and chromosome Y haplogroups. So it was possible to associate in this population that they have a more concentration of markers from European ancestry, but they have also about 20%. There is a variation. You can see the big deviation from this box from African ancestry and also for Native Americans. And that's different in the same level from the controls that are not so different. But when we went to the chromosome Y haplogroups, this haplogroup R1b, it's carefully associated to European ancestry, so there is a big association with this European ancestry and haplogroups from type 1 diabetes. We also look for HLA on those patients, and again, we can found these haplogroups associated of DRB1, DKA1, and DKB1 associated with the previous described haplogroups in Europe, O301, O301, and O201, and again in patients with O301, O501, O201. So these DRB103 and DRB104, when they are present, they are in heterozygosis or homozygosis. They are much more frequent and associated to the European background and also to the R1b haplogroup of chromosome Y. So this is insisted to us that there is probably an influence of the European background in type 1 diabetes, but as we are admixed people, those marriages are also giving to these descendant this haplogroup and conferring also risk to admixed people. So we look with this possibility to also check questionnaires that are sent to many of these individuals of their background. They have answered a questionnaire about their relatives in the first grade, second grade, and third grades, where they are from, if the parents are native in Brazil, or if they came where they came from Europe, Africa, or where else, and also how they declare themselves as white, black, parda, or indigenous, and also from Oriental background. So in Maranhão, we had this conclusion that R1b haplogroup is associated with DRB103 and DRB104 alleles, and there is more than 50% of ancestry in the European side. So for this study of the relatives of the Brazilian, not all these 1,700 people responded to the questionnaire. So we had about we have 662 people that answer the questionnaire and have information about the region of the where they are and about their classification as auto-declaration, what from race and skin color, and where they live, and where from the parents, grandparents, and great-great-parents. Of course, most of the people do not know exactly where from their great-great-parents and where the race color of their great-great-parents, and a little more knows about their great-parents, and of course they know some their parents and grandparents. So we had seen that from different regions in Brazil, there is more influence from Italy, from Japan, from Europe, but most of the Brazilian came from Peninsula Iberica, also from the European ancestry are clearly marked in these people, but we have also Midwest and Africa, and but there is some difference on the second and third degrees, but not from the those that are born in Brazil and born outside Brazil. So we have investigated in the allele group level because we have few numbers of these individuals, so we have to condensate and explore at the group level of HLA. And as you see, there is a multitude of color in these graphics and this information, but sometimes we have this kind, there is much less variation in the allele group, in the people that declare themselves as indigenous, as we know that there is an association to population that are not mixed, that they have a less repertoire of HLA groups. Also, people from Oriental level, they are also less represented with colors in the graph. So here we see the indigenous groups and also the yellow groups with less colors and much more well represented from some allelic groups from also DRB1 and DKB1 and DKA1. So most frequent allele in patients with T1D were those that have already been described as risk alleles in multi-center studies conducted in Brazil and other countries. So as we know, DR4 and DK3 are the most common alleles associated with type 1 diabetes. Both genders reported as being from admixed or brown people. The relatives' birthplaces were in Brazil since they are here from beginning of this century, and only great parents came outside from Brazil, and they are from the Peninsula Iberica, and which reflects the Brazilian history of colonization. And as you can see, DRB103, 01, 0501 for DKA1 and 0201 for DKB1 are the main haplotype. So we can link the ancestry of patients with T1D, diabetes type 1, back to the European and Caucasian ethnicity. But we also have to explore more this as a conclusion of this paper. So we had the chance to had more people typed for HLA. So at the moment that we have 1,600 patients typed with different methods, as Dr. Ennis demonstrated to us, with all of medium to high resolution. We can input DKA1 by the linkage disequilibrium. So we have at that moment this information, and we test for hard Weinberg equilibrium. That is very important to know if there is the population was equilibrated, and we also confirmed some of these ambiguities by NGS typing, and we done this imputation for some of this ambiguity based on self-reported color, race, and region algorithm. So we had almost the same number of male and female people of much older than 20 years old. So it was possible to compare in the future with these individuals that we use from the Brazilian Registry of Bone Marrow. So it was also possible to compare. We see later, and we have the association with the onset, people that started with diabetes very early, before 4 years, between 5 and 9 years, different proportion, and different population from different regions in Brazil. The North region, it's very few people, only 45 for these typings, and conclusions of the North regions are not so strong as others. And also we concentrate our efforts in white people that self-declare as white, admixed or brown, and black. As you see, people that declare this as Orientals from Asia, and indigenous are very few people in this group. So as can you see, there is a big concentration of European, but there is a variation, and people from Native American also are represented, and African origin. So if you put in the main cross here, we have a vast majority of predominance of European ancestry, but also there is some Native American and African American genomic areas in these autosomal chromosomes. And not for different surprise, they are reflected in the very large number of alleles that we have detected. Of course, that is a bias, that is a disease. So O301, it's very important, as we expected. They are far from what is found in Brazil studies of other populations, or in the Brazil bone marrow registry, and also for 05, 04, 05 for the RB1, and there is also association with the key A1, 03, 01, 05, 01, with much more important figures than in the Brazilian population studies. and also for DKB10302, that is also known that is largely associated with type 1 diabetes. So, but when we took for a PCA analysis, they sought out in a certain way some alleles that are much more frequent in the European ancestry, those that are also frequent in this group, but due to admixture, they share some alleles, and also from Native Americans. So it was possible to calculate risk haplotypes. So there is a gradient and a percentage of risk haplotypes with the DRB1, O3, O1, O5, O1, and O2, O1 for DRB1, the DKA1, and DKB1, with a very important figure, and then O4, O5, O3, O1, and O3, O2. And there is, among those people, also protective haplotypes. We still study if those haplotypes are associated with some best prognostic in complications, but for age onset, we will see later that there is some very important risk haplotypes and protective haplotypes associated with the onset of the diseases. So this is the last paper that I will show the results for you. We had the opportunity to look on these 1,600 typings, and the information about the age of diagnosis. We grouped the individuals less than 6 years, 6 to 11 years, more than 11 years to 19 years, and more than 19 years. They are well balanced people, and they are also, except for the North region, also sufficient numbers to generalize the information about regions. And also we also pick up the information of if they declare themselves as white, brown, black, Asian, or Native American. So in this comparison, we set information that there are differences among those peoples related to gender. Of course, the groups are separated by age, and by age of the onset of the diseases. And there is no big difference of the socioeconomic status about, but not everyone had answered this question. And of course, there is also a self-reported color race that is very different about this group. It's no different in genetic background. So the alleles that sought out as protective for age onset of the diabetes, as you know, those people start with the appearance of autoanticorps, and then there is a decrease in insulin production. And countries that have a very screening the population about type 1 diabetes, as the Finland and Norway, already know that some alleles that are associated with the onset of the disease. So for us, it was important to give a better healthcare assistance if we know, as in Finland or in Norway, if they had already an allele that will mark the onset of the disease. We can give information, precautions about some lifestyle, and then prevent or delay diabetes type 1 onset. And this is a mark conclusion of those countries in the North Europe that already showed this case. So there is a protective alleles as the RB10701, 0602 for the TB1, 1303 for the RB1, and 0102 for the KA1. And as we also imagine, the KA1, 0301, and the KB1, 03, 02, and the RB1, 04, 05 are the major risk for those risk alleles. We think about how those haplotypes will be expressed in the surface of these cells. And thanks for Dr. Anis to their previous presentations. Here we have only class 2 in the surface. So we can imagine if they have just one haplotype, one allele in the haplotype, or 2 of the risk alleles in the haplotype, or 3 of the alleles in the haplotypes, we have a different response on the onset of the disease. And of course, if they are homozygous of this certain allele, or completely the 2 haplotypes of the risk allele have the 2 alleles set. And of course, we can have all the combinations, and some haplotypes that didn't sort out on the in expectation maximization theory to give rise to the, as we don't know the father and the mother haplotypes, we use a sort of program that is very well known in the HLA literature and platform, that is Harlequin, to infer haplotypes by expectation maximization. But here we made this calculation, and we explored this combination, and we sort out 0301, 0501, and 0302. As of course, there is a very important risk we well know from the previous study, and it sort out also the 04, 05, 03, 01, 03, 02 that is already mentioned, and 04, 02, 03, 01, and 03, 02 as risk for the onset of this disease. And then not for a big surprise, that is already previous depicted in some studies, that this haplotype, it's very common for the disease risk, but it's not associated with the onset of the disease. So 0301, 0501, and 0201, it's a risk of the disease, but all groups have this haplotype. And not only people with 0 to 5 years, 6 to 10 years, 11 to 18 years, or more than 18 years. So you can see that it's about 45% in the first group and also 32% in the last group. So it's a mark of the disease, not for the onset. But on the other haplogroups, 04, 05, 03, 01, and 03, 02, we have these haplotypes going down. So the red mark, it's going down. So it's really associated with the onset of the disease. And also 03, 01, 501 and 0302. So those are haplotypes that are risk for the onset of the disease. And when they are associated, as you know here, about 40% are associated with this haplotype of the mark of the disease. So there is another role of protection or association of the disease with some haplotypes of the presence of some alleles. So we have this question not answered, is still not answered in this group. So we continue to further do some analysis. So Professor Gomes is still arguing us with these common haplotypes in African, Amerindians, and European in the Brazilian population. And some are from the European ancestry, some are from African ancestry that are present in this population, in the type 1 diabetes population in Brazil. And some came from Europe, some came from, are still present in Brazil, and if they from Amerindia are protective from diabetes. But we are still doing some research. Now we are looking for SNP associations, and of course there is a much higher interest. There are some SNPs associated with retinopathy and peripheral nervous system diseases. So we are going to present our preliminary results in the Congress in Brazil this week. So we hope we have this information consistently compared to the Brazilian HLA to do a manuscript in the next month. So I will let the message that in Brazil we have this facility that we have the Brazilian registry of bone and marrow donors, and this is a very good source of information, so we can compare this frequency of these alleles and haplotypes. And we have recently sorted a CEDD allele frequency map for the REDOM, and can be used to compare for this association of diseases. So with that, I could not emphasize the role of Professor Armanilia Gomes and her previous PhD students that I mentioned in some of the studies, and Professor Negrato from Bauru and Campinas, who is also a leadership in Brazil from type 1 diabetes. The entrance in the group of Rosana Azulay from Maranhão that has this control group and gives some opportunity to compare with a direct population study. And my colleagues at the LDD lab at UERJ, Professora Daisy Silva, Lívia Ferreira and Paulo Ricardo are now studying specifically mitochondrial DNA, Lívia, and chromosome Y for Paulo Ricardo. And also people in my lab that to do this typing. Some are not working more in the lab, but I should mention. And this work was done with support of CNPq, the Brazilian national agency, and also from our agency in Rio de Janeiro is the FAPERJ. And the partnership with Inca and Redomi with Dr. Daniela Oliveira. Thank you very much for your attention.
Dr. Porto, thank you so much for your presentation and congratulations for your work. It's not easy to develop a multi-center study, and especially with genetic information. I mean, because of actually it's really expensive because for this kind, because we had to say it for HLA testing, we need to work on a high resolution level. So it means sequencing. And it's really hard to implement because it's not easy to analyze the data. It's a really big data analysis. So you need to work not only with, biochemists, immunologists, also with bioinformaticians. So it's a really hard work with a lot of professionals involved. So I really want to say congratulations because it's just really hard to develop that kind of research.
Thank you very much. It's
Oh.
really expensive, and fortunately, at that moment we had a Brazilian with a real dollar very opportunistic time, and we could at that moment bought some reagents at a very good price, and so to do this typing that is very expensive. We cannot see very commonly 1,600 type in a disease situation. It's more cheaper to do GWAS analysis.
Yeah, definitely. And well, it's something that it's a challenge here in Argentina, like in other countries in Latin America, Africa. So I think it's really important to face the truth here that we can do science, we can do a really high level and multi-center in research, but sometimes it's hard because we unfortunately got that kind of problems that it shouldn't be, but we have it. So but it's important that we share that we share that we can do it and we are able to develop that kind of technology in our countries. So really, thank you so much for your presentation and for showing us your really hard work. If is there any question from the audience, please unmute yourself. And you can also write it on the chat. I can read it for you. I would like to ask you, well, first of all, it's really important to share that this kind of data analysis not only shows medical information. I mean, we all more like biology medical background, but it's also important for develop other kinds of studies. I mean, social and anthropological studies. So how this information shares with other colleagues in another fields that are also important to analyze the data. It's not just from a biological point of view, I think. And also how important it is to know or knowing who we are, right, as a population, as we people. So I also want to notice that I would like to ask you, because you share, I asked Dr. Anis about that, if they get in Pakistan, like a bank or a database from the country. I know, well, Argentina and Brazil, we usually work together. So I know about the databases in Brazil, but maybe you can share with the audience how you guys work with that, because it's a really important database and genetic back that you develop in the country.
So we have this facility, Brazil, in the beginning of 2000, started with this registry, and we have this facility, and it's a good sensibilization and solidarization of the Brazilian people. And now we have about almost 6 million people that had been typed. And since 2021, we are now typing with NGS, so the information in the 5 loci typed in the information. So the paper that I show you is also the information after 3 years that we have this NGS typing on the Redom database, and it was with 300,000 individuals typed. So every year we put more 100 and 150 new individuals coming into this database, and we are preparing to have this information of haplotypes, and this information will be very useful for not only for transplantation, the right people to the bone marrow transplantation, but also for other solid organ transplantations that we can predict in certain ways, finding a possible match for kidney transplantation in the region, so we can look for in another region and things like that. So we are very excited to produce this information, and we have this facility also that we have a very strong association in Brazil with histocompatibility and immunogenetics, and we done regular annual events, so people are in contact, and so we share. And Inca, with the presence of Dr. Oliveira, is very confident that we have to share this information, and that will allow us to do more predictive and precision medicine. So it's really an opportunity for us here.
Yeah, 100% agree. So thank you so much for sharing that with the audience, because maybe we are in the field, so we know about it, but not everybody knows that. So that's why I really think it's really important to take these meetings to talk about it. So from your presentation, I would like to ask you again, if it's any question from the audience, please unmute your microphone or write on the chat. I would like to ask you, when you mentioned about the HLA-DR and HLA-DQ alleles on diabetes, but they are more related to predict association with diabetes, but not with complications of the disease, right?
Yeah, for now we have not sufficient number of people to really ascertain this association. There are some alleles that sort out, but when we do the correction with the Bonferroni, it vanished. So there is some kind of flag, but I cannot say correctly that, so this allele will be associated to people that will develop a kidney disease or a peripheral nerve disease or a retinopathy. There is some complication on the interpretation of this data. As you know, we are now exploring diabetes type 1 as for some individuals, some patients, it not only is go with a singular disease, it came with other autoimmune diseases. So we also taking to analyze if those other autoimmune diseases in patients that have type 1 diabetes appeared first, appeared later, and what are the alleles that are shared or provoke or associated with these grave diseases, and their shared DRB3 allele. So sometimes they have a common allele that provokes autoimmune disease, and why one developed a disease in the pancreas and the other in the adrenal, it's not so clear to us. Some previous infections, not only the genetic background could be the key.
Yeah, well, that's really important what you're saying, because maybe I'm generalizing. It's not the same for everybody, of course, but for diabetes or type 2 diabetes, it's more like a silence disease. It's more like a laboratory finding. But in type 1 diabetes, usually in children, it's more like the absence of the presentation of the disease is really sometimes suppressive. Like you got a patient who lost conscience in an urgent care in the hospital. It's a really traumatized situation, not just for the kid but also for the family. So this kind, the HLA typing is not something that you used to take into consideration for now in clinic. So I would like to ask you if we should implement this in patients who are diagnosed with diabetes type 1, but not just on patients, also in the related, because they can maybe get siblings, so they can share some of the HLA genes, and maybe we can help to prevent that kind of episodes in the related, I don't know, cousins, siblings, father, mother, yeah, also, because again, we think about diabetes type 1 in children, but it also can happen in adults. So I would like to ask you about that, your experience about testing, more members of the family, not just the patients.
Yeah, there is a big question that we also discussed here. It was very expensive, and it's expensive today. So the problem or the solution is that it's being more and more accessible. So not only it was very difficult to set up a lab that with all the infrastructure and to do that. Now with NGS and the new machines of NGS, the price to do sequencing is going very, very dramatically at the same level to do exome of HLA typing for. So why to do exome? Why not to do exome and HLA typing? I think it's a moment that we are living now to give information. But for general purpose, for healthcare ministry to decide to implement this, we have not a suspicion and not access to many diseases that are from not well-developed countries. So sometimes it's very difficult to convince a ministry that it's an investment necessary. Our dream is that we can, like in Finland, could do in the we have in Brazil the food test that do the innate metabolic test for babies. So we can put these markers in this test and then already know for almost for the family that know that they have a type 1 diabetes, do this test, and then it will be cheaper and give an information. But it is still a dream.
Yeah. Well, it also happens, I guess, with other immunological diseases like inborn errors of immunity, that it really took time to add that on the neonatal screening, for example. So yeah, I think it could be a really interesting tool, especially thinking now that I'm talking with you to you, thinking about maybe, yeah, it's expensive because we need sequencing, because we need a high resolution testing, but maybe it could be also, it could be implemented some other test with, I don't know, low resolution test. And if you see some of the alleles there, you can go on for a higher resolution test only on patients where you could actually detect it, those specific alleles. You know, like maybe have a first screening technique, low resolution, and then go with a higher resolution.
I totally agree. I totally agree.
Okay, so I don't know if anyone in the audience got any other questions. So yeah, that's a lot of comments about thank you and congratulations for the presentation. Again, once again, Dr. Porter, thank you so much for your presentation and congratulations for your work. So
thank you.
Thank you for being here. So let's move forward with our next speaker, Dr. Daniela Fernandez Soto. She's a biochemist specialized in immunohematology and blood banks. She's a member of the Laboratory of Histocompatibility at at Garrahan Hospital here in Buenos Aires, Argentina, and the Public Umbilical Cord Blood Bank since 2010. So Dr. Fernandez Soto, thank you so much for joining us today.
Thanks to you. I'm going to share the slides. Will you see?
Yeah, we can see it now.
Okay. Sorry.
No, it's okay. Yeah, we don't see the full screen. There you go.
Oh.
There you go. Yeah, now we can see it. Thank you.
Okay. Good morning, everyone. I'm very pleased to be here today to present our study from the laboratory titled HLA Diversity in the Argentinian Umbilical Cord Blood Bank, Frequencies according to donors' reported ancestry and geographical distribution. First, I'd like to thank the organizers for giving us this opportunity to share our work. I'll try to speak slowly so that everything is clear. It's a bit a challenge for me to speak in English for a full hour. During this talk, I'll guide you through the main findings of our study, showing how HLA diversity varies across ancestry groups and regions in Argentina. I hope this will give a clear picture of the rich genetic variation we observed in our cord blood units. Before we move to the main results, let's briefly cover our disclosures. I'd like to note that we no conflict of interest to disclose. Let's start with the basis of HLA. Now, as we know, the primary role of HLA molecules is to present antigens to T lymphocytes, triggering a specific immune response. It's also important to highlight that HLA molecules are codominantly expressed, which means that both alleles of a given gene are displayed on the surface of cells at the same time. Understanding this fundamental role of HLA is key, as set the stage for interpreting the diversity patterns we observed in our study. With this background, let's now explore the implication of HLA diversity in more detail. In addition to the codominant expression, HLA molecules exhibit an enormous degree of genetic polymorphism. This diversity gives us an evolutionary advantage as a species, helping our immune system recognize a wide variety of pathogens. However, there is a flip side, because HLA molecules are highly monogenic. This same diversity can become a challenge for individuals needing a transplant, as finding a compatible HLA donor is much more difficult. And it's precisely this fascinating challenge that motivates our work and brings us here today. Let's look beyond function, and let's look at their geographic distribution. When we analyze the frequencies of HLA alleles across different regions, we find significant variations depending on the population and the area studied. For example, here we show a distribution of 4 DR beta alleles worldwide. As we can see, each allele is common in a specific region and may even be completely absent from others. These patterns highlight how HLA diversity is shaped by population history and migration, which is critical for understanding both evolutionary dynamics and donors matching in transplantation. The Argentinian registry, known as INCUCAI, was established in 2003. Today, it includes over 4,000 cord blood units and over 310,000 volunteer donors, more than 90% of whom have been typed at high resolution level. From this registry, 712 donors have already participated in donation procedures for both Argentinian and international patients. Moreover, this national effort has made possible 1,912 unrelated donor hematopoietic stem cell transplantations for patients across Argentina. It's so important to note that previous reports have described HLA diversity in Argentina's population without considering the country ethnic or geographical variations. Wait a minute. No? The population of Argentina, is the result of intermixing between indigenous, American, and European and African groups. An interesting previous study used ancestry informative markers, or AIMs, to see how this genetic contribution varied across different regions in the country, and to compare them with what people reported about their grandfather's origin. In this graph, each bar shows percentage of European ancestry in blue, indigenous in red, and Africans in green. As you can see, these proportions change from one region to another, showing how historical migrations and admixtures shape the genetic mosaic of Argentina. With this background, our aim was to describe HLA diversity of bank umbilical cord blood units collected across the country. We used next-generation sequencing technology to report HLA alleles and haplotype frequencies, taking into consideration donor-reported ancestral origins, as well as geographic distribution. Let me tell you more about Argentina. So, Argentina has a very diverse genetic background. Most of our populations came from European immigrations, mainly from Italy and Spain, but also an important contribution from Native American groups like Mapuches and Collas, as well as previously from Africa and Asia. Now, this diversity isn't uniform across our country. In fact, people with Amerindian ancestry tend to live in the north and the south, while European descent are much more common in the central region. For this study, we collected 451 umbilical cord blood units from 6 provinces. Most of them, about 70%, came from the central region, which you can see in blue. And the rest were from the north. Oh, sorry, about 7 came from the central region, which you can see in blue. And the rest from the north, highlighted in red. Here. When recruiting donors, we asked about their ancestry. Following HLA net recommendation, we grouped into 4 categories. South American, of European descent, people born in South America of European ancestry or European living in Argentina. South American of Amerindian descent, people with Native American ancestry, mixed ancestry between Amerindian and European population, and other or unknown ancestry including Asian, African, or any combination of these with Amerindian or European origins. What's interesting is that both European and Amerindian groups were almost equally represented. We could see here 171 versus 184. And as we expected, European ancestry was more represented in the center, while Amerindian ancestry predominated in the north. Let's move to the results. Here we can see the HLA allele frequencies across all loci for class 2. For class 1. Both of the total population and European and Amerindian ancestry. For HLA-A, we found 42 alleles common. 6 were quite common, together making about 2/3 of the total, and 2, 0201 and 2402, stood out with frequencies about 10%. Interestingly, 12 alleles appear only once. As expected, HLA-V shows 88 alleles. The most frequent were B51, B18, and B4403, while almost a quarter of these alleles were seen only once. You could see here. Finally, HLA-C include 34 alleles. 34 alleles. 8 covered about 70% of the total, and 3 showed frequencies over 10%. We ran the same analysis from European and Amerindian groups to see how this diversity plays out within each one. Now, let's look at class 2. For HLA DR beta 1, we identified 47 alleles. 5 of them were relatively common, making up about 35 of the total of frequency. The most frequent alleles was 0701, followed by 0301, while 9 alleles are rare, appearing only once. For HLA DQ beta, 17 alleles were detected. Interestingly, just 8 alleles account for almost 89% of the total frequency, with 3, you could see here, standing about 10%, and only 3 alleles appear just once. So again, we see a pattern similar to class 1. A few alleles dominate, while many are rare, contributing to overall diversity. Let's go deeper into allele classification. To explore our HLA data in more detail, we classified alleles according to the common intermediate and well-documented catalog. For HLA-A, most alleles were common. We found 4 intermediate alleles. For HLA-V, 10 alleles were classified as intermediate, as we can see in this part of the slide. Well-documented alleles were also observed across all loci. Interestingly, some alleles typically reported in other populations appear in our groups. Like these alleles highlighted with an arrow, the first, this, or 104, usually seen in Asian Pacific Islander populations, appear in our European ancestry group. And the second, common in European and African population, was found in Amerindian group. So even though most alleles are common, we also see these interesting signals of ancestry-specific or well-documented appear in an unexpected group, highlighting the rich diversity of our population. Moving to HLA-C and class 2, for HLA-C, we identified 28 common alleles, 2 intermediate, both in the Amerindian group, and several well-documented alleles, mainly associated with European and Hispanic populations. Notably, some of these were detected in an unexpected group. Within these alleles for class 2, we observed 43 common alleles for DR beta 1, 3 intermediate, and 1 well-documented, known for European and Hispanic ancestry, found in other groups. For DQ beta, which is less polymorphic, we found only 50 common, 1 intermediate, and 1 well-documented allele. The later, this 104, 02, 03, the later usually associated with European and Asian Pacific Islander ancestry detected in Amerindian group. Overall, this analysis confirms that most alleles are common, but the presence of intermediate and well-documented alleles, sometimes in unexpected ancestry groups, reflects the complex of HLA diverse landscape in our population. Let's look at some standout alleles in class 1. Among the common alleles, A6817 has previously described and is frequently found in our population. This allele was first reported in the Colla amerindian of northwest Argentina. At low resolution, A68 was present 65 times in total. With this allele family, A6817 was seen 21.5% of cases. That's 14 times. Now, what's really striking is when we consider ancestral origins, the allele appears 7 times in Amerindian groups and only 2 times in European group. Further analysis of these units, we notice it usually appears in combination with alleles in B locus, also describing our populations, such as we can see here. Moving to the other alleles that also show difference between both groups, these alleles show was found 8 times in Amerindian group and 2 times in European group, while O222 appear exclusively in Amerindian group. This highlights how ancestral origins influence allelic distribution. Now, let's shift to B locus. Here we can see an allele commonly found in Amerindian population from Mexico to Argentina. Appear 12 times in Amerindian group and only 2 times in European groups. Other alleles previously described in population from southwestern Brazil and northwestern Argentina were also seen more frequently in Amerindian groups. Let's turn to class 2, which is our focus on HLA-DR. Building on those ancestral patterns, we saw an HLA-A and B unified similar trends here, really emphasize how Amerindian influence shaped this distribution. Analyzing this, the DR B locus, 3 alleles previously described in Argentinian Amerindian populations particularly caught our attention. What's noticeable, and I think it ties back beautiful to what we discussed earlier, is that several other alleles also had a marked predominance in Amerindian groups compared to the European groups. It really underscored the impact of ancestral origins on histocompatibility profiles. In summary, so far, we can say that the present study on 451 high-resolution HLA-type cord blood units revealed distinct allele frequencies variations based on donors' reported ancestral origins and geographical distribution. Building on this, following this initial work, we expanded our analysis by comparing Argentinian HLA genotypes using the Aplostat database. For each sample, we applied this tool against global reference populations with particular focus on the South or Central American Hispanic group, analyzing unphased genotypes. Now, let's see the haplotype results. Here, I'm simply showing the graph summarized information obtained on HLA haplotype frequencies in relation to various global and ethnical specific populations. What I'd like to highlight is that out of 451 umbilical cord blood units analyzed, only 420 showed frequencies in one or more of the reference populations. But what about the missing ones? Among the 31 not represented in the Haplostat database, about 7.2% of the total, only 7 were reported as having a European ancestry, and all of them were collected from the central region of Argentina, obviously. Below, we can see 7 haplotypes highlighting the alleles classified as intermediate, well-documented, and some common. We believe these alleles might be underrepresented in the database, even though in some cases we also observe uncommon CB and DRTQ linkage disequilibrium. And now the Amerindian units. Within these 31 units not represented in the Haplostat database, 14 units, the 45%, were reported as Amerindian descent, and 12 of them were collected in the northern region. The remaining 10 units had mixed or unknown ancestry. 2 were mixture of Amerindian and European ancestry, and 8 had other or unknown origins, 6 of which include some Amerindian component. For these units as well, we have highlighted the alleles classified as intermediate, well-documented, and some common, which observe our attention. So, what this means? All haplostats includes HLA haplotype frequencies relative to various global and ethnical specific populations. Our results show that our ethnic minorities are still underrepresented. Therefore, further analysis of HLA frequencies will help improve collection policies in the public cord blood bank to achieve its goal of increasing our patients' chance of finding compatible grafts. Now, let's move to the discussion. When we tie everything together, so far, we've seen pattern in HLA alleles, but why does this matter and how can we use this information in practice? In our laboratory, we are fortunate to have access to high-resolution HLA data, both from umbilical cord blood units and from some patients, a true gold of information, true gold mine of information. This allowed us to improve the clinical impact for our patients, as knowing which allele are most common in our population greatly facilitates our work in the lab, enable more accurate and reliable interpretations. The next two slides will illustrate this in practice. This knowledge help us better characterize alleles and haplotype frequencies in our populations. And here's our ultimate goal. In the future, these frequencies could even be applied to solid organ allocation, for example, by calculating the panel reactivity antibodies, improving donor-recipients matching and patients' outcomes. Finally, because our population is underrepresented globally, it's also essential to identify which regions of our vast country are optimal for recruiting volunteer donors and cord blood units. I show you an example shortly to highlight why this is so important. Let's move to the next slide. Here's a real-world example. This example highlights why it's so important for HLA laboratories to know the alleles frequencies in their population. We typed a family at intermediate resolution and observed an unusual result. HLA-A 6806 appear in both the mother and the patient, a rare allele in our population. Upon reviewing these reaction patterns, we suspect a proof issue, specifically with proof 321, which may misidentify more common alleles for us, such 6801 or 6817. Interestingly, when the samples were tested using different reagent kit manufacturers, this problem did not occur. We also examined samples with previous high-resolution results for 6817 and 6801, performed SSO typing to evaluate the proof. For this analysis, we conclude that the proof fails to detect A6817, while it correctly identified 6801. And we can see in our daily routine, this type of issue occurs frequently. For affected sample, we adjust the cutoff for the bid in the analysis to prevent miscalls and ensure accurate HLA typing. Another example from the laboratory. This example also highlights why knowing our population's allele frequencies is absolutely essential in our work. Using a sequence-specific primer typing kit for HLA analysis, we found a false positive result in well 12, for example, with, guess which allele, the HLA 6817. High-resolution studies confirm this. Just like the previous example, this kit also misreact with A6817, but not like with 6801. Without knowing HLA 6817 is common in our population, we wouldn't have spotted this issue. So, allele frequencies knowledge is a key tool for accurate results. And with these two examples, it will be clear that in Argentina, we have alleles HLA 6817. Now, let's talk about solid organs. As we know, CPRA is a game changer in solid organ transplantation. It's essential for fair and efficient organ allocation, and it precisely measures a patient's immune sensitization, helping us predict how likely they are to match with a donor. But here's the key: to calculate it accurately, we must know our population's HLA allele frequencies. Why? Because these frequencies determine the weight of each antigen in our populations, allowing us to tailor organ allocation to our country's genetics reality. This approach reduces rejection risks, improves graft and patient survival, and ensures a more equitable distribution of organs. Countries that adopted calculated PRA achieve better clinical outcomes and greater transparency. So we must know our frequencies. Here's a real patient's case. This is a clear example of why we should seek to better represent our ethnic minorities. As part of our work, we assist in unrelated donor search for patients needing hematopoietic stem cell transplant, and we often face cases like this one. We present a real patient currently seeking a compatible donor. The slide shows in red box the high-resolution HLA typing of the patient and all donor options from the World Marrow Donor Association network, which now includes over than 44 million donors worldwide. Since donors typing differ in resolution, type low, intermediate, or high, we sometimes need to evaluate the population background before deciding whether to propose a potential match. In this case, the patient carries HLA-V 4013, first described in Argentina, and the R-beta 1 0407 allele, common here, but from a highly polymorphic family. None of the haplotype, including B4013, are listed in Haplostat. The orange boxes show the probability of the donors. If we contacted again and type a high resolution, we'll have 0, 1, or 2 mismatch. Given our unit transplant policy to accept only 10 out of 10 or 9 out of 10 or haploidentical donors, the best option here would be to proceed with an haploidentical transplant rather than continue with international search. So, what next? I think perhaps if we had more voluntary donors from our ethnic minorities, we could find a suitable match for cases like this more often. For this reason, further studies will help us to improve recruitment strategies across our country, aiming to represent all of our genetic diversity. And this work continues. We analyzed 451 cryopreserved umbilical cord blood units, all typed at high resolution. Currently, 600 additional units are under analysis. This ongoing work reflects our dedication to understanding our population immunogenetic profile. By expanding and refining this knowledge, we aim to optimize clinical strategies and enhance patients' quality of life. None of this would be possible without our amazing HLA team, and I'm truly proud to be part of it. We extend our sincere gratitude to all collaborators, donors, and institutions that made this research possible, including the INCUCAI Registry. Special thanks to Valeria Roca, our CONICET researcher, who drives our daily effort to make this work visible. Thank you for your attention.
Thank you, Dr. Fernandez Soto, for your presentation. I really think it's, again, like we discussed with Dr. Porto, the importance of knowing who we are, the importance of developing this kind of research where it's not only showing us how we can work on a medical science field, but also on migration, anthropological, and social analysis. And they are both related, because sometimes we think that maybe biological science is not related to social science, and actually, yeah, it is, and it's needed for also treatment of pathologies like transplantation and how we involve to or how we work to improve the treatment of the patients. So
It's
thank you so much. Yeah,
because when work in the laboratory daily with patients, we haven't time to analyze and to dedicate. And I think it's important to us to know our frequencies. We have the luck to have people that investigate, and maybe we try to publicate, but in old days we couldn't. Well, there
Yeah.
are a lot of things that we had a pediatric hospital with a number of patients important, and sometimes we thought we must to publish this, we must publish, but the time isn't infinite.
Yeah, that's why, 100%. That's why we talk about translational medicine. I need to work together as a colleagues with clinicians, with researchers for doing this, because it's not just the day by day in the clinic and the medical attention for patients. It's also for doing research, and that takes time. And it's impossible for doing only one person, only one professional. We need a team.
Yeah. I'm lucky if I had a good team to work. We expected publish the other work in nearby. See, nearby.
That's really good news. We're waiting for reading your publication. That's really good news. So is there any question from the audience? Again, you can write it, type it on the chat. I can read it for you. You can unmute your microphone and ask Dr. Fernandez Soto whatever you consider. I would like to ask you, Dr. Fernandez Soto, because I think it was really important that you share real life scenarios, something that could happen in the lab, both. I mean, if you're doing research, but also if you work in a hospital with clinical care patients, how you can manage those scenarios where maybe the results are not really frequent if we used databases, international databases that sometimes are not related to diversity in the country where that test is performed.
We work in a public hospital, and this is an advantage because if we suspect, we could repeat the sample, we could investigate, and now the registry could study our samples if we need at high resolution level. All of this help to our suspect in the daily, but I think that the HLA works need to find which alleles are in our region because it's really, we see A6817 all regions, I think. So we have more patients from the north in our laboratory. The people came from our hospital, and we see examples like this. We see frequently another B39, I think, 39, and others. We have travels. Sometimes the manufacturers are not interested in change or investigate our alleles because the manufacturers are made her kits for his countries especially, includes and when made kits that includes alleles extra has a cost, extra cost.
Yeah.
And it's a problem because the costs are higher.
Yeah, and especially, well, it also goes on the quality of the results because, yeah, when you work on transplantation, you may usually get access to the family members, so you analyze the entire genetic haplotype of the family. Not always you get access to all the members of the family, of course, but it may help you to analyze and to be more accurate about your result. But it's not always like that. And also, we are talking during all this meeting about HLA typing in other contexts, not only transplantation, also disease-related or HLA disease-related scenarios. And in that case, you got only the patient because you're not going to analyze the family members. And of course, if you got only one data from one person, it's really hard to choose, especially if you're working, because that's what usually happens. In the clinical field, you work with intermediate technology or intermediate testings. So it's really hard to analyze and to realize which allele will be the one you're supposed to number or you're supposed to identify as your result, because in intermediate testing, you got a group of alleles. And of course, everybody knows about the limitations of the technology. Yeah, but if you already know the allelic distribution in your population, it's easier to choose which allele from that group will be more accurate to the patient, and that will be also an increase the quality of the result you inform, and that's
to the analysis.
Yeah,
Patients, for the transplant patients, the analysis for when monitoring the antibodies is also interesting, because we have the transplant organ, the organ solid transplantation, as we know, as you said, at intermediate resolution, and we sometimes suppose which allele of this could be, and we analyze if the patient had this allele, this antibody could be a donor-specific antibody, and all this knowledge is necessary to our population. Not if we typed wrong at first time, we couldn't could not follow the patient post-transplant correctly. Isn't good. It's important. Yes.
Yeah, because the result, it's a directly clinical impact on the following, or we can say on the outcome, clinical outcome of the patient. So,
yeah. It's important.
Really, yeah, 100%. Because again, it's a clinical outcome of the patient. Yeah.
And so I insist with know where our minorities, ethnic minorities are, because we found all the time in search for patients who need a transplant, bone marrow transplant, we saw every day, now we have 3 or 4 patients that had no options for unrelated donors, because his alleles, their alleles, are not common in other populations. Sometimes we depend on Brazil, or sometimes from Chile, but our country, especially the north and the south, are not represented well. And I think we have a mixture of ethnicities that is important to know where we must to collect it more volunteer donors from our registry. I insist with this because I saw daily in our lab, and it's important also.
Yeah, 100% agreed. That's a really strong message. So thank you so much for sharing with us your hard work, your daily work. Really, we really appreciate that. And so again, if as you can see in the chat, there's comments from the audience. Thank you and congratulations for the presentation. So this is the end of the course. I hope everybody there enjoyed this opportunity to learn from really experts in the immunology and transplant immunology field and HLA-related diseases. Thank you so much to Dr. Rumbo, Dr. Anis, Dr. Porto, Dr. Fernandez Soto. As you can see, we try to organize these presentations with the following presentation complement the previous one. We started with immunology transplantation, how cells and the autoimmune response are involved in transplant immunology. And then at a molecular level, when we talk about HLA molecules, they impact not only on transplantation, but also on HLA-related diseases, and how this is important to analyze in a higher demographic study, how we can use in a practical way this information in several scenarios, and how and the impact of this information in health and our patients. And again, also as a social and anthropological studies, how important it is to know who we are, where we are from. And so it was really an honor to have all of you here. You got a really incredible CVs, several publications. You're directors of several doctoral theses and research projects. So thank you so much for taking the time, for sharing your knowledge with us. We are really happy to have you here, and it was really an honor. So thank you from the Argentinian Society of Immunology and from Immunopedia for joining us today. So if you let me just a few minutes, because some of the audience were interested about Immunopedia's work, we would like to tell you that we wait for you if you would like to join us as an Immunopedia ambassador. At the Immunopedia website, you will find information about not just transplant immunology, also other fields in immunology like infection, cancer immunology. So we really encourage you to share with us your research topics if you are a professor or assistant professors on college, and you will also find their information, quiz, something you could use for your classes or lectures. Also, if you are a student at college and you need information for taking your classes, for your exams, you can also find their courses, you can find opportunities. Let me share just a little bit. This is a flyer of a talk that I will present on the 4th Franco-Argentine Immunology Congress that will take place on this Wednesday, October 29th to this Saturday, October 1st. So in this conference, again, the Argentinian Society of Immunology organized this annual meeting. So if you couldn't be able to participate this year, we encourage you and wait for you for joining us next year. On this Friday, October 31st at midday, we're gonna talk about Immunopedia. So if you can share this information, because the target audience is residents in immunology, students on college, PhD students or postdoc who really wants to know more about careers opportunity, how you can apply for scholarships or courses and international workshops that Immunopedia organize, as well as different tools you will find in the website. You're welcome for joining us in this talk. Also, again, if, like I said before, if you would like to become an ambassador of Immunopedia, you're more welcome to join us and to work on science communication. That's something that we really encourage from Immunopedia all around the world. So please, if you can spread the word with your students, with your residents, with your PhD students and postdoc, you're more than welcome to join us this Friday. And you will find all the information on Immunopedia's website. I will share with you again. You will find it here. This is the Immunopedia website. So if you would like to contact us, send an email, you will find it there. And also I will share again the Argentinian Society for Immunology website. It's in English and Spanish, so you will also find information for the next meetings and events that the Argentinian Society for Immunology organize. So once again, thank you very much for all the audience, for all the participants to be here. we really hope this is the very first meeting from so many others in collaboration. So thank you so much for everybody. So see you next time. Have a really nice day.
Thank you,
Bye.
everyone.
Thank you very much.
Thank you, Alejandra, for all your
Bye.
efforts.
No, please, thank you, Bon, for making this
Thanks,
happening.
Lee.
And also Natalia Satucci. Thank
Thank
you.
you, everyone.
Bye.
Thank you, everybody. Thank you.
Thank
Bye.
you.
Thank you, Dr. Anis.